Okay, going live. Let's hope this works. Good morning, afternoon, evening, wherever in the world you are. Very, very warm wishes from slot 2 of the webinathon of the International Year of Basic Sciences for Sustainable Development. My name is Arnab Bhattacharya. I realize that may be a little tricky for many of you. So let me just put it on the chat over here. And I'll be taking you through the next 4 hours from 9 o'clock UTC till 1 a.m. or 13 hours UTC. We are based in Mumbai, India, and we've lined up a wonderful series of talks and discussions, etc., involving speakers from all over the world. Before we get there, let me briefly tell you about basic sciences and why I feel they're super important. We've all heard certainly about Einstein and about his theories of relativity. Well, most people probably don't know that we use these every day. Our mobile phones, which have GPS receivers in them, which we use to book a taxi or call something or find out in maps where we are, wouldn't work for more than 2 minutes if the GPS system did not correct for relativity. Now, Einstein didn't come up with the equations for relativity 100 years ago saying, oh yes, 100 years in the future, somebody might want to book a taxi. Let me come up with relativity. No, he was just exploring the world, finding out the laws of physics, trying to discover something about the world around us. Similarly, we've all been through this COVID-19 pandemic. And thankfully, this was a case where vaccines were developed very quickly, and within a year we had them. But behind this is years and decades of work in basic sciences. How do you get that vaccine inside your body? It's carried by some little, a little bundle, a liposome. And people worked on it not knowing that, oh yes, 40 years later, somebody's going to use this to deliver a vaccine during a pandemic. We all use the internet. You're watching this on the internet. The internet was not developed because people wanted to share videos. It was because high energy physicists at CERN wanted to share data. So there is a huge, relevance for basic science in today's world. And today, June 5th in particular, is a great day because it's the World Environment Day. On World Environment Day, it's a time to step back and look at the status of this beautiful planet we live on. Unfortunately, this planet faces some rather serious problems, especially with the environment, whether it's global warming, deforestation, groundwater depletion, you name it, our environment today is in trouble. And again, if we need to find solutions to save this planet, this can only come from the basic sciences, which is why the UN and other bodies are celebrating 2022-23 as the International Year of Basic Sciences for Sustainable Development. And you're watching this 24-hour webinathon. Now, of course, it's going to get nothing so serious. It's going to be fun things. And the first thing is, let me show you a 1-minute clip of a teaser of this IYB SSD. So let's share screen. Okay. I hope it's visible. Okay, that was a very short clip. Before we start this slot 2, there's one thing I'm supposed to let you know. That's the periodic table challenge. So let's take a look at that. We'll first watch a video, and then I will tell you a little bit. There's a small correction, and we'll also take a look at the site. The representatives from the IUPAC will be monitoring all the responses they receive during the next 4 hours, and at the end, they will probably declare some winners. So let's hope this works with this time with the sound. Again, let me share this. Sound should be okay. All right. Okay, so that was a video. There's one small change. So let me just show you that as well. The only small change is that the phrase IYBSST, that word must be added to the city field. That's where you add it. That's the only change that there is. Otherwise, you can go ahead, just search for it. So don't worry, you can enter anything in school institution, but make sure IYBSST is in the city field, and then you can play this game. And it's really easy. You go to the website, just Google IUPAC periodic table challenge. It's really easy. You can play it multiple times, and that's going to be how it is. Okay, so I hope that gives you something interesting to work on. And we hope there are many entries during this particular 4-hour window that we have, and there are nice prizes at the end. So let's do that. So as we come up to 9:10 UTC, or it's 14:40 in India, it's a pleasure to get started off with the first of the talks we have lined up today. And this talk is going to be given by Professor L.S. Sashidhara, who is presently the Center Director of the National Center for Biological Sciences, which is probably India's premier laboratory for biology in Bangalore, India. And he is going to talk to us about how the science of life makes for a better science of life. You need it. And I'm going to pass it on to him. He will introduce himself and then go on with the talk. I know that if you're watching us on YouTube, you can't ask us questions, but please be sure to put in your questions and comments in the chat. I'll be monitoring the chat and I can read those questions out for Professor Sashi. And we have some other volunteers who are also looking at the chat. So if you're watching on YouTube, please enter it in the chat. You can enter it in the chat also in case you're on Zoom. Let's go ahead with this, Sashi. So why don't you spend a couple of minutes, introduce yourself, and then go on. I will spotlight your video and you can take over from there.
Thank you, Arnav. Can you hear me?
Spotlight. Yes, we can hear you, Shashi. Absolutely, we can hear you.
Okay, thank you very much. Good morning, good afternoon, good evening, whichever the part of the world you are. And it's a pleasure to be here. It's an honor for me to be here. And as one of the speakers of this 24-hour the webinar-thon on the importance of basic science in our day-to-day life. It's being a biologist. Arnav did mention that I'm a director of a biology research institution. Obviously, I'm a biologist. So much of my talk would be more in the area of biological sciences. Before that, just brief introduction about myself, which is right here. I'm a geneticist, developmental biologist, and also an evolutionary biologist. But obviously, evolution is something, a common thread across all of biological sciences, across the scale and complexity of the field. I did my PhD at Cambridge and worked at multiple places in India and currently at Bangalore, at the southern part of India. My main interest outside research is in the area of education, particularly undergraduate education and pedagogical innovations. And I'm also interested in public health, mostly because of the pandemic, I got into this area. But earlier, prior to pandemic and even now, I'm generally interested in science administration, policy and strategy, and internationalization of science. And I'm also part of multiple, organizations and various different initiatives. And internationally, I'm a fellow of European Molecular Biology Organization and immediate past president of International Union of Biological Sciences, which is a partner to International Year of Basic Science for Sustainable Development, along with other unions. With this introduction, I'll go back to my talk. The title is somewhat tongue twisting, I'm sure. More science of life for better science for life. As what I do is going to sort of discuss some of the fundamentals of biological sciences, as discovered how they have helped to improve the quality of life in general, and both at the conceptual level and also at the level of how we live ourselves in a society along with other very diverse group of humans as our neighborhood. And being a International World Environmental Day, obviously it's also important that we learn from what we have learned in biological sciences to improve the quality of our surroundings, particularly not just the physical parameters like pure air, comfortable temperature and so forth, and timely rainfall. It's also important that we sustain biodiversity. And modern understanding of biology, particularly ecology and evolution, has immensely helped the very different ways we can conserve and expand our biodiversity around surroundings. Unfortunately, we need bigger commitment from entire world, every citizen and every society, every nation, every government, to improve the presence of biodiversity on Earth. So I'll start with very simple few examples, basically tracing the history of biological sciences, how the very understanding of life itself is helping understanding ourselves. As you all know, most people will ask this question about who am I, what am I made up of, or what is my relationship with my surroundings, whether it is a physical surrounding or the biological surroundings. And in of course, human surroundings too. But human surroundings, you always give certain names that I belong to this community, I belong to this nation, or belong to, I'm a male or a female, I'm a younger person or older person, and so forth. But is more importantly is very existence of life on earth and very existence of human life on earth. We always ask this question. Philosophers have asked, and every citizen, the person could be a farmer in a small rural place, or a person could be a corporate, CEO, ask these questions. So how we have addressed this question over a couple of 100 years, in the past 100 years, 200 years or so, I just am going to give an outline. Very quickly, I'm sure all of you have heard the Gregor Mendel's work, and you also have read in your school textbook or later, or even in newspaper, some of the work that Mendel did, and we call him as father of genetics. He studied the patterns of inheritance, from how traits are inherited from parents to offspring. The bigger picture he was answering is not simply how a few morphologically identifiable characters are inherited. Bigger question he was asking is how we inherit everything that constitute life. For example, what is born to a pea plant or to a human parent, a couple of human parents, is another human being. First, it is another living organism. So what is inherited is the life in itself, all the features to make life. So the patterns of inheritance that Mendel discovered helped subsequently to understand the chemical basis of life, because then you start looking for what is being inherited, right? And that basically led to the discovery of DNA, which many of you have heard, and I'll come to that in a minute. In parallel, although they didn't know about each other's work, Darwin was trying to understand life somewhat differently. Darwin was not simply looking at, what are the different types of organisms that are present on Earth and how are they adapted to their local environment. To some extent, or he was able to answer those questions in the immediate terms. But the long-term impact of theory of evolution, as proposed by Darwin, is helping us to understand the very origin of life and its diverse forms in which we see today. Which also, when it's coupled with the Mendel's work, which will actually help us to understand various different aspects of the existence of life on Earth, and later the very different processes which are involved in maintenance of the living condition as against the non-living state of matter, right? All of us are made up of the same matter. I don't have to repeat here. We are basically, the bottom of everything is physics, and there's the matter that, all the laws of physics that govern the matter, the same laws of physics govern the matter in our body, except that they're manifested somewhat differently, and what we call as, the life. And trying to understand that part is, which basically happened in the mid 20th century or second half of 20th century. But the beginning, the conceptual understanding for this and the trigger of different way of thinking about life started with the Gregor Mendel and Charles Darwin. Now, in fact, in Darwin's book, the Origin of Species, the last paragraph, there's one sentence or a phrase. He clearly mentions that perhaps there is a single origin of life. Today, looking at the chemistry of life across all organisms, it's really clear that at least there could be multiple different origins of life. What exists today as basically from one single origin of life. All others must have become extinct in case there are multiple origins of life. Again, I don't have to get into the details for any audience of anywhere in the world. There is something called chemistry of life, right? Now, there is something, what is called DNA is the genetic material, and RNA is an intermediate messenger where information that is coded in the DNA gets converted into another form of nucleic acid. And from this, the information is decoded in the form of protein. It's almost like saying that there is information written in a textbook or in a book. You read that information. Perhaps you make a copy of that information basically, let's say, a temporary copy of this. DNA is a permanent copy. And that temporary copy, like RNA, is used, and so you send it to another place, let's say a factory or somewhere. You say that, use this information, can you, fabricate something for me? And what is fabricated is a protein. And the protein, depending on the structure, depending on certain chemical properties of protein, will have certain specific function, right? And that basically is the what you call as the very central to life, the chemistry of life, right? Now, this is similar across all organisms. Irrespective of whether you are a bacteria or a plant or any form of animal or a human, the DNA is exactly the same chemical entity, and how information is replicated, or how DNA is copied itself, the process of DNA replication, what we call, is a chemical process, is exactly the same. Similarly, when RNA is made, the process is exactly the same, and then the protein is made, right? And this process is called transcription and translation, because simple, it's a very, interesting, fun explanation to this process, but it's a deep meaning to it. Transcription means you have taken the information to another script, but the information in the same language. Translation means information is translated to another language. Similarly, nucleic acid is a form of language, and amino acid is another form of language. DNA and RNA are nucleic acid language, and protein is an amino acid language. Now, most important information that both Darwin and Mendel were looking at, how are we seeing these differences and why there are differences, right? If there is a particular form of life, why there is multiple different forms of life? What is the core to understanding the biodiversity, core to understand the human diversity, core to understand the differences within the family, is to understand where the source of variations and how these variations interact with the environment, right? So interestingly, the chemistry of DNA is such that the very chemistry of DNA, that is, it has nucleotides on two strands, and the nucleotides on the both strands are complementary to each other. They make certain kinds of chemical bonds we call as hydrogen bonds, and very strong bonds. And because of this, they are very stable. Even at 90 degrees centigrade temperature, DNA is stable. Only when you increase the temperature to 95 and above, it starts breaking down. In fact, there are bacteria which actually survives at even 120 degrees centigrade. There are certain mechanisms to keep the DNA, well protected. Coming back to this, but sometimes there can be a small mismatch that can happen. But because DNA replication is a particular process called semi-conservative replication, that means the two strands get separated, and strand A becomes a template to make a new strand. Strand B becomes a template to make another new strand. So at the end of this one, we'll have two copies of DNA, but each copy will have one old strand, one new strand. Because of this, if there is a small error in the DNA replication, like what you call as mismatch, it gets carried to the next generation, right? That is your source of variation. Interestingly, it's purely stochastic event. You're right. The chemical, bonds that are formed is because of the collision of atoms. I'm sure all of you know about it. And typically in the biological system, there is a very clear, over the time, what has evolved is a system in which these collisions are made more deterministic. For example, in the air, for example, sodium and hydrogen may be combining together and making an ammonia molecule, but that the quantitative level at which it happens, whether it happens or not, it's so rare that you will not smell ammonia, in your, simply in your bedroom or in the living room. But in the biological system, these collisions happen in a very organized state of matter that is present in the cell, right? Because of which it happens at a much more deterministic pattern. So, but if the stochastic reason, A, for example, is supposed to go with T, A may go with C, right? But that is tolerated for a short time. Otherwise, the stability of the DNA will not, will be lowered if there is a mismatch. But for some of these, one generation or so, it is tolerated, such kind of a mismatch. And that what carries over as new variations. By the way, I'm very consciously using the word variation, not mutation, because mutation in general public has a negative connotation. Mutation is always considered as something wrong, something negative. But variation is simply another variant. There are, in the context of viral pathogen like SARS-CoV-2, a variant we call is a mutation, and we consider it as something bad because that will affect us and may cause diseases. But the variation can also be beneficial even in the context of post-pathogen interaction. For example, the Omicron variant that came out in the end of 2021 turned out to be a boon because it spread widely. At the same time, it was not causing the disease and generated more immunity among the population. Whereas the Delta variant was a deadly variant, which basically caused huge number of deaths across the world. And this process, when it's reiterated, is going on and on for the last 3,150 million years, or what you can call as 3.75 billion years. And what you see on a National Geographic, channel is the kind of biodiversity or different life forms that we see. The most beautiful ones are all, irrespective of what they are, every one of those organisms are extremely beautiful. And the source of variation, their interaction with the environment, is basically what helps to explain not only what is life and also all forms of life that exist on Earth. So the integration of Mendelian genetics and Darwinian evolution, what has helped us to understand this. Now, one of the other things we also learned in basic science is without variation, there wouldn't have been a life immediately after the very first form of life was born or originated. It's nothing like born. But born is when a life is born from another form of life, we call it as life, is birth. But otherwise, there is the origin of life. Origin of life happened at 3.75 billion years ago. If we were, let's say, static, there was no variation in the DNA replication, there was no new variant, that form of life would have become extinct on the same day because the environment is continuously changing. Environment changes because of the dynamics of the Earth and also the solar system. Environment is also changing because of the variation environmental perturbation that living organisms generate. For example, oxygen went to the atmosphere. Now we have 23% of oxygen in the atmosphere because of this, the reactions that are happening in the living systems. So variations are extremely important. Now, diversity is key for survival. Without population level diversity, in any form of organism, bacteria to human, you would not have survived as a species. What survives, what perpetuates is a population, although it may happen at individual level, because the environment will have a sweeping impact on all individuals, and what survives is the ones which can survive in a given environment. So variations are very important. Making a population homogeneous is actually not useful. So one of the other things we also want to know is, in which way it will help to know our own evolution. Now we have sufficient evidence on human evolution. We also one of the branches of the Darwinian tree of life. And in that one, we and chimpanzees form one branch, and chimpanzees we separated at two branches within that branch. And only about 5 million years ago from a common ancestral great apes. But human evolution is not a single step. There have been multiple steps, multiple species evolved and became extinct, and only survived is currently the Homo sapiens. Based on the fossil evidence, based on the dating, we know that there's so many species evolved, and many of those species also exist at the same time and space, in the past. For example, if you look at in the 0 to 1, about first couple of hundred, 0 to 1 is basically 100,000 years ago. Millions of years ago, there was a time where 3 to 4 human species existed at the same time and space. For example, Neanderthals and Denisovans, we existed at the same time and space as recently as 30 to 40,000 years ago. Not only we lived together, we also met with each other, we mated with each other, and in our genome, we actually carry the Neanderthals or Denisovan genomes. That means human species is not something called, what people call as pure species. We are just part of any other animal, and we went to the same process of evolution of a species, giving rise to human. Now, the next part is, fine, we all understood about the biological basis of our existence. We know a little bit about the matter, like DNA, RNA. There is something called self, which we consider as something which is conceptually very different. some people use the word consciousness, some people use words very different other names. I do not get into the religious or politically different names associated with self. You simply understanding being aware of myself. Let's say you consider that understanding the self. So people have actually taken this forward very well using very different animal models. For example, like two insects, they behave differently depending on the context, depending on their body size, depending on their status of hunger or something, suggesting that they have some understanding of the self, and accordingly they behave differently with other individuals. For example, if these two individuals are fighting, if something consecutively 3 or 4 times loses the fight, it stops fighting with others, right? It has certain understanding of self. There are also very different experiments to suggest that that to some extent it has some understanding of its body size too. The great example is chimpanzees. People have shown that chimpanzees can actually recognize themselves in a mirror. There is no only other animal other than human which can recognize itself in the mirror as far as we know is chimpanzees. And we considering that they all have reached certain levels of cognitive abilities, but humans have certain additional abilities because of the language that we have evolved, and that language has helped us to pool our thoughts and experiences. So for example, in all other animals, the experience of self, what drives them to do something different. Others cannot learn innovation that happens in its neighborhood. We are the only one across the time, whether it happened 2,000 years ago, 3,000 years ago, we continue to use, because the way we can explain to each other with the help of modern language, the cause and effect relationship, very different things that happen around us. Now, coming back to this one, variations in the DNA copying mechanism is the most important aspect of chemistry of life, and which basically helps us to generate these variations and very different environmental ones, which is also equally important for our survival. Because of this, we also have, I'll skip this in the interest of the time. People have realized that, irrespective of, for example, if every individual is different, right? So, then how do you say someone is superior, someone is inferior? So everybody should have, certain kinds of, equal rights in the world, right? Each one is a different variant. Some may be visibly different variants, some may be not so visibly different variant, but everybody should have equal rights. Now, as you can see here in the 21st, particularly late 20th century, the early 21st century, the more and more acceptance of human diversity and more and more, vocal support for equal human rights across the world. This is something which is a conceptual, contribution of science to our life. I'll take a couple of examples how it has actually helped in our material life, right? Now, whenever, Arnav gave an example of 100 years ago, what Einstein, proposed, helped us to, develop this mobile technology, which we are using, all of us are using. Now, sometimes it takes very slow pace. Sometimes it can also be real time. So I'll give 2 examples. Slow time scale in the biological sciences, this one. When it took about 30 to 40 years after, Darwin and Mendel to discover the physical basis of genetic material, what is called DNA. And also what is the functional material which is associated with very different chemical processes called enzymes or proteins, right? For example, insulin is one such protein which was discovered in 1920s and which is involved in glucose metabolism. And as soon as it was discovered, people realized that we can use insulin, and also at the same time, Darwinian evolution actually helped us to understand that these physiological processes in glucose metabolism are more or less identical among all mammals. So people started extracting insulin from slaughterhouses and give that as a medicine for diabetic patients. While there was a cure available now, a treatment available now, but it was very expensive because only a small amount of insulin was extracted from the slaughterhouses. But later, with better understanding of the chemistry of DNA and very different technologies that helped us to make recombinant DNA technology, what they call. So you can now take insulin DNA from human and put it in bacteria. For bacteria, is simply another piece of the same chemical molecule that it already has. So it undergoes DNA replication, DNA to RNA transcription, and RNA to protein translation. But what comes out is a protein called insulin, which is useful for glucose metabolism in human cells, so human tissue system. So you can take that insulin and give it to patients. Now the cost is so low. Now the entire world, the insulin treatment to diabetic patients, wherever insulin treatment is required, is very affordable to the whole world. This is another example. This happened over 100 years of time. It's a slow process from Darwin Mendel to giving insulin, which is made in bacterial cells. But to look at what we had just experienced the last 2 years, the basic science was used every day, and also whatever we learned in the past was used to address this pandemic, right? Within days, within hours of whatever the new discovery happened on which virus, how it enters our cells, how it can be stopped, and where the different variants are being generated, which may become more aggressive, which may become less aggressive, which may spread more, which may spread less, how to test, how to isolate. All kinds of things were exchanged in real time because of the technology that we had. And that helped us to, prevent this pandemic within, about 2 years of time, because of the vaccine that were developed and very different drugs. And finally, it has come to a stage where WHO itself has declared that this pandemic is over. And just one or two more examples. I have another 4 or 5 minutes. I'll sort of quickly take you through, because I'm coming from India. India is one of the largest, it is currently the most populous country in the world, 1.5, 1.6 billion people, right? And when India became independent from British colonial rule in 1947, is one of the poorest of the poor countries. It had just come out of famines, and the whole world had come out of Second World War. So there was so much of hunger and poverty in India, and that's the time when Indian and those leaders of that time decided that let's use the science the way to provide the solutions to the real life problems. And indeed, we invested heavily, even in the poorer state of country. We invested heavily in science, basic science and technology both. Perhaps India is the only country which invested in basic science so much in such a state of poverty. And that basically helped to become self-sufficient in food and nutrition. Now, actually, India is one exporting food and donating food to other countries. And the kind of mathematics and physics that we practiced, it helped to do the kind of space research that we're doing, satellite technology that we have accomplished, and of course, the IT revolution that came out of India and also spread to the whole world. Chemistry and chemical engineering was also well supported in India, and which helped India becoming the largest pharma industry in the world. It's actually the supplier of low-cost medicine to the entire world. Same thing with vaccine and biotechnological production too. So India is now the 4th or 5th largest economy in the world. It all happened in just about 75 years, and starting with a very small number of scientists and bureaucrats, because they are the only literate people in those days. Even literacy was very low. And people always think that basic science should be nurtured and protected, and should be protected and nurtured. And people think that basic science is only a luxury, and we can only develop countries can happen. But basic science is universal, and it should be nurtured in every corner of the world, because the talent can come out from any place. Even under extremely constrained conditions, people work. Right? It has been shown in Europe also during Second World War. Some of the major discoveries were made by basic scientists. Even in India, people have shown that under constrained conditions can do. For example, Raman spectroscopy, which every industry uses, every lab uses these days, all over the world. It has every hospital we use, was based on the work that Raman did under extremely constrained condition in Kolkata in India, before independence in 1920s, using that an equipment which he built from a donation that he got from some people. Other people in the recent time, in the last 30-40 years, Shambhunath De, Dilip Mahalanobis, Ramalinga Swami, these are the people who did work which has actually saved millions and millions of lives across the world. The rehydration therapy, ORS, what they call, or which is treating the diarrhea. The entire Latin America, Africa, Southeast Asia, Asian countries, everywhere it has saved millions. In fact, Ramalinga Swami's work on iodized salt is basically taken to the whole world, whether it's developed or underdeveloped countries. Plant tissue culture, the whole world knows about it. It's actually the seed was, the work was seeded in India in 1950s, and which has now become $1 trillion in the industry across the world. It all started with the basic science and understanding the process of de-differentiation in plants. Plants are the one, unlike animals, they can actually, a cell which is differentiated can go back to its original state called de-differentiation, and can give rise to the whole plant. So going lastward, India now Earth is a global village, and we are all connected, a click of a button in real time. And interestingly, when human evolution happened, and we are a small group of people who are in Kenya or Eastern Africa, and then migrated to multiple places and got isolated. That's why we have so many, distinct sort of communities in Africa or Australian aborigines or Papua New Guinea people, separated by 30, 70, 80,000 years of time. But now all isolated communities are connected because of technology, right? But the problems are also becoming global. Climate change is a global problem, right? We need, pandemic is a global problem. We need, global science to solve those problems, but the solution is somewhat more local. So each country, each region has to look for its own innovative solutions for the local, the problems of, climate change or a pandemic, because the impact will be different in different parts of the world. So science has to spread to the whole world. So as you can see here, we should have more basic science every part of the world, more interdisciplinarity, because more and more scientists have to come together, coming from different, disciplinary background. And there should be more internationalization of science. And in scientific information, it should be peer-reviewed. Scientific information should be available, and data should be available to everyone. Otherwise, people will rely on unsubstantiated information and irrational solutions to their problems. So it's very important that we share, valid information across the world. I'll stop here. Thank you very much. I'm happy to take some questions.
Thanks a lot, Shashi. That was a very nice, amazingly diverse tour through many centuries of biology, and into some of the very interesting things that happen in India, right from ORS solutions for dehydration to iodized salt and a lot more. Wonderful talk. The session is open for questions. Let's quickly check if there are any questions on the YouTube channel. Let's see there. Can I get the YouTube channel link? Okay. Okay, there is one question on YouTube. I will just read it out for you, Shashi. I hope you can hear me.
Yes, sir, I can.
Okay. It says that one of the problems seems to be plastics or microplastics in water. Are there any technologies available, easy, low cost, perhaps? I think the person posting is from India, to detect plastic in water.
Yeah, of course. I mean, technology is already available for many of these things, right? But only the problem is the scale at which we want to, deploy such technology. It's not affordable. In fact, some microplastic or from soil or from bacteria or from marine system to detect, at least detection is possible. And at the same time, when removal, there are certain technologies available, but those technologies are not affordable yet. That's where more and more scientific discoveries have to be made to make such kind of technologies affordable, so that across the world we can scale. Because the scale of the problem is so large, no technology is feasible to deploy to solve that kind of a problem. But at the same time, people are using the basic science methodologies to address some of these problems. As you know, the world has gone through very different unlivable conditions, extremely high temperature, extremely toxic chemicals deposited simply because of the natural process. Those toxic chemicals are mostly inorganic chemicals, not necessarily organic, is simply because of all kinds of heights, and then the ice age and everything. But the life has continuously lived for 3.75 billion years because organisms have evolved over time, slowly, right? And of course, the stochastic process of variations, which simply the scale at which these stochastic events are happening, generated sufficient diversity to develop mechanism to adapt to these conditions. So now people are now accelerating the evolutionary process by generating more and more variations at a larger scale of bacteria or other such microorganisms. Hopefully at some day we'll be able to, people already have some sectors that practice degrading bacteria, right? So even microplastic is also can be, subjected to bacterial degradation. So that could be a good solution and long-term and a scalable solution.
Yeah. Okay. Any other questions? Let me just check again. No. Okay. Any questions on the Zoom? Okay. Actually, I had one observation. I mean, it was nice when you. It's also interesting. I'm just picking up from your iodized salt thing. Even the entire the tests we have today for the thyroid hormones, for thyroxine, etc. A lot of it is actually, I mean, yeah, today, of course, people make tests because they want to detect a particular thing, but a lot of it arose because of very basic research into the structure of these molecules that people were interested in. So I think there is really an enormous basic research that underpins a lot of what people consider medical advances in the world today.
Exactly. No, I can give you another example. Everybody knows these days, RT-PCR test, whether they know what goes behind RT-PCR test or not. Every citizen of the world has understood that there is something called RT-PCR test, which can be used to detect viruses and get themselves either isolated or happily move around with the people. Now, that RT-PCR is basically based on the chemistry of DNA and the chemistry of RNA and the process of transcription. And there are also people later discovered when we are working on the certain viruses cause cancer or trigger cancer. They also found out that there is a reverse process where RNA can be used as a template to make it's a DNA copy. The information can be converted back to DNA, and that is we call as reverse transcriptase. So RT-PCR basically reverse transcriptase driven PCR or reverse transcription PCR, right? So this is used in basic science all the time. Not only it was a product of basic science, it's also a methodology used in more discoveries in basic science. The first time in our humanities history, such high-end technology was deployed at a scale globally to address a pandemic, right? And if there were no RT-PCR test, there were no way if you are asymptomatic and still carrying the virus, and you would be spreading to everybody. Another 4 or 5 years would have continued to have this pandemic.
Right. I mean, I guess it's something. Okay. I think, Narendra, if you have a question, please go ahead. Please unmute and ask.
Yeah, thank you, Professor Shashidhara. I just would like to know that NCBS is working in many areas. Can you elaborate a little bit in the context of school students and teachers that what sort of sustainable development activities through NCBS are conducted?
Yeah, so in the context of sustainable development, there are two.
So maybe we should just remind people, because we have listeners from everywhere, that the NCBS is the National Center for Biological Sciences in India.
Okay.
They may have forgotten it from the introduction half an hour ago.
Yeah, I know. Yeah, sorry. So, it's like in many, top quality research institute all over the world. NCBS also is dedicating itself to use basic science for sustainable development. And the sustainable development means it should be sustainable for a very long time under very different conditions of the future, which we cannot even predict. To understand that, you need to know better. Mechanism should be known even better than giving a short-term solution. For example, if you want to cure cancer, right, you need to know, you have to have a deep insight of cancer. If you want to simply treat it such that a person can somehow sustain for another year or so, you don't need to know all about the cancer. You give certain kind of treatment and you stop at that point, and that person will survive at least for a year. So any problem, you need a long-term solution, sustainable solution. You need to know better the mechanism. So in the context of nutrition, right, how you can produce very nutritious staple rice, right? You need to understand the biological mechanism, very different nutrients that are produced in and under different conditions, stressful conditions. For example, The rice grown under certain stress conditions may be depleted with certain kinds of nutrition. You may still have certain amount of starch, but that's not, producing only the crop in terms of starch requirement. Is also about varied nutrition. So one aspect of our research is related to that one, using the genomics approach, understanding the process by which very different nutrients are produced even under stressful conditions, so that which can be transferred to one or two high yield varieties, so that we can produce nutritious rice under heavy rainfall condition, low rainfall conditions, high temperature and all conditions. The second is the biodiversity and conservation related. With better understanding of the genomics and ecological interaction with the different animals and kind of thing, we are better prepared now to recommend certain kinds of conservation, practices to the forest officials, which you are already doing, and more of it will come very soon, and as the years pass.
Okay,
Thank you,
great.
Professor.
Thanks, Shashi. Thank you for participating on this
Thanks for the
Slot 2
opportunity.
program. And I think it is it was really nice to hear all your thoughts. I hope the audience also appreciates it. Remember, you can still put in your questions and comments on the chat. We will be going through it periodically, and we can always go back and get answers to these questions from Professor Shashidhara. So thanks again. Thank you very much. And let's move on. So I realized that I had a little, I made a glitch while sharing the video. I forgot to share the sound. So since we have about 6 minutes to go before we have to hand over to the next panel, I'm just going to play the periodic table challenge video once again. So let me just do that. Where is my periodic table video? Okay, that's the window to share, and let's hope I get it right. I will share. I have to share sound, optimize for video clip. That is the window. Share the window, and let's hope this time. Somebody please tell me if the sound is not there this time. Please put it in the chat so I know about it.
Hello, everybody. You already know the periodic table of the elements, where all the chemical elements are grouped into columns and rows. But how well do you know these elements? I propose you to take the periodic table challenge, an online quiz that is produced by the International Union of Pure and Applied Chemistry, the IUPAC. Your challenge will be to answer 10 randomly chosen multiple choice questions about the elements. As any effort should bring a reward, you will first get a certificate from IUPAC if at least 60% of your answers are correct. But there will be more for this special edition at the occasion of the 24-hour online event organized by the International Year of Basic Sciences for Sustainable Development. Among the winners of today, we will randomly select 3 who will receive a poster of the periodic table signed by 13 chemistry Nobel laureates. You can play alone or with a team from 5 a.m. UTC 5 June to 5 a.m. UTC 6 June. So, how do we play? First, go to the homepage at iupac.org. You can just enter periodic table challenge in your favorite search engine. You will certainly find it easily. You can choose your favorite language, and then here, your difficulty level. Once done, you have to choose an element, which will be your avatar. Let's go. Now, be careful. You have to enter your name, and here in the school institution box, type IYBSSD so that you are sure to enter the special edition competition. Do not forget your email, your city, your country or region. Then send. Are you ready? Click on play, and here is the first question. And if you do not reach the 60%, you can just play again. But do not forget to follow also the very interesting talks, panels, discussions, interviews, videos we are proposing to you all along these 24 hours. We hope you are enjoying this event.
Okay, I hope I got it correct this time and you could hear the sound as well. Remember, there is a small correction. The correction is that please put your city as IYBSSD. The city is IYBSSD. Doesn't matter what you put in the school or institution, but don't worry, I'm sure the IUPAC people who are monitoring it will be very happy to give certificates to anybody who has IYBSSD in their thing today. Let me also, since I have a minute before I have to hand over to Sizun, let me see if I can play the teaser video again, which I missed again. Okay, just a second. We play this one. Full screen, and I hope now the sound of this video is there again, the teaser video for our International Year of Basic Sciences for Sustainable Development. So as the video says, Basic Sciences provides the tools for sustainable development. Okay, so it's time to get going, and let me see if, yes, let me add spotlight over here. Okay, so before I hand off, we're going to keep the theme of a little bit of biology going, and I'm going to hand off to Christian Sizun from the International Union of Pure and Applied Biophysics, who are going to be having a very nice set of small talks over the next hour or so, and she will coordinate it. Sizun, I think I have made most people co-host. I will be monitoring the thing, and if there's anyone of your speakers, I will make them co-host, so there's no problem with screen share. And so I am handing it over to you with about 2.5 minutes of extra time to go. So we are on schedule. So all yours, all yours.
Thank you very much, Arnab. So welcome everyone to this session, which is called Biophysics for Global Health and Sustainability. And this is organized by the International Union for Pure and Applied Biophysics. So my name is Christina Sizun. I work at the French Center for Scientific Research and I represent IUPAB, and I will share this session. And so I will share the screen. I hope this works. I hope. Okay, I think it does.
Yes, your screen is visible.
Okay.
You might just want to go to full screen mode.
I hope I try this again. Is it better or not? That's always the problem. Not really. This will also be the problem for others.
Maybe you can first go to the slideshow and then share the screen.
Yes, I will do that. Oh, it's the same problem. Okay, now I still have, and so this is where the 2.5 minutes go. And technical issues. It seems that I don't get it.
Okay.
Is it better now?
It's still the same thing for me.
Okay, I don't manage. I don't know why.
Or just share your desktop and do it or whatever.
Okay, is it better?
Yes, yes, now it is.
Fantastic.
Now we have full screen.
Okay, thank you, Arnab. So
okay, and see,
this
you're
is
right
not
on time
about
now.
science, but about technical issues. So yes, so welcome to the session. I will very briefly introduce IUPAB. IUPAB is the World Organization, a federation of organizations in biophysics founded in 1961. And its aim is to coordinate and support research and teaching in biophysics through several activities and scientific events, and more particularly to support young scientists in this area. So before we hear about our panel, I will take a few minutes to introduce you to the concept of biophysics. Indeed, biophysics is standing apart since it is a hybrid science, and biophysics is a bridging scientific discipline in which the theories and methods of physics are applied and developed to understand how biological systems work, and this at different scales ranging from molecules to whole bodies. Biophysicists are working within a variety of disciplines and fields, mostly centered on biological science, but they are also at the interface with chemistry, physics, engineering, mathematics, and computer sciences. Biophysics emerged mid of the 19th century when physicians concluded that living systems obey the same universal rules as inorganic matter. And 1 century later, physicists and chemists started contributing to life sciences by bringing cutting-edge tools that permitted to reveal atomic details of biological molecules and structures inside of cells. Biophysics have been critical for building many tools, and new tools are still developed. And I will briefly review some contemporary biophysics tools. These include spectroscopic and microscopic techniques for structural biology. And these tools are based on quantum physics and classical mechanics. Very recent techniques include free electron lasers that allow describing very fast motions in proteins. Cryo-electron microscopy that gives high-resolution 3D structures of very large protein complexes. And here we see the coronavirus spike protein, and this allowed to understand how antibodies bind to it. And the last technique here is cryo-electron tomography that provides direct insight into the cell interior. Computational modeling is another aspect of modern biophysics, and artificial intelligence is now very efficient in predicting protein structures. Molecular dynamics are calculations that are used to model biological systems like proteins or even viruses in interaction with membranes, and this at the molecular level. Biophysics further includes tracking single molecules and cells and membranes in real time, as can be seen here on the left-hand panel. This is done by combining optics and mathematical analysis. And last but not least, biophysics includes multi-scale modeling of organ systems. For example, modeling of cardiac function by integrating information at different levels, from the molecular level to the full body, and using here electrophysiology and biomechanics. So biophysical tools are critical to present-day research to unravel really complex biological questions, but they also underlie many applications in modern medicine. For example, biophysics has given us imaging using radiation technologies like magnetic resonance imaging, computed and positron emission tomography scans, these for disease diagnosis and therapy. And the physical principles behind this are the interaction between electromagnetic radiation and matter and magnetism. Biophysics has also given us the development of new devices, and this can be kidney dialysis apparatus, defibrillators, pacemakers, artificial heart valves, and all of these are sustaining life and also based on physical principles like electricity, magnetism, or rheology, that is the study of flow properties. Biophysics has also given us biomaterials and biomechanics, and for example, allowed to invent novel materials for prosthetic limbs. It has also given us nanobiotechnologies that are used nowadays for modern DNA sequencing, drug delivery, and the detection of pathogens. And biophysics has also given us environmental biophysics. These consist in tracking and modeling our Earth from the stratosphere to deep ocean vents. It allows us to track and to monitor pollutants, and also it provides us with new sources of energy, like turning algae into biofuels. And again, there are physical principles behind this, like thermodynamics, spectroscopy, quantum biology. So in summary, biophysics has established a way of quantitating biology. It has pioneered inter, multi, and cross-disciplinary science, and this is really important that many sciences work together. It provided a breeding ground for new methods and approaches, and I summarized some, and it produced foundations for medical applications and for environmental causes. So it is now my pleasure to welcome a panel of 4 renowned scientists who accepted our invitation and who will talk about important questions that they address through their research in the framework of biophysics for global health and sustainability. Our first speaker will be Matthew Higgins. He is a professor of molecular parasitology at the University of Oxford in the UK. His research team deciphers the molecular basis for host-parasite interactions in diseases such as malaria to guide the design of vaccines. Our second speaker is Pimchai Cheyenne. She is a professor of biochemistry, professor and dean of the School of Biomolecular Science in Rayong, Thailand. And her research team has contributed to understanding the mechanisms of different enzymes used, for example, in biocatalysis and synthetic biology. Our third speaker is Jesus Perez-Gil, professor of biochemistry at Complutense University of Madrid in Spain. His work is focused on pulmonary surfactant biology and lung homeostasis, and ranges from fundamental research to establishing diagnostic and therapeutic tools. And our last speaker will be Miguel Castano, professor at the Faculty of Medicine at the University of Lisbon in Portugal. And the aim of his work is to unravel the physical principles that determine the interactions of peptides and proteins with lipids that are related to viral fusion, so to viral infection, and to develop new drugs. So we will now listen to Professor Higgins. I will unshare my screen, and so you can share your screen. So please, Matthew, the floor is yours.
Great, thank you. You can see the slides there.
Excellent.
It's a great pleasure to be able to speak to you today. I'm going to tell you about how we use structural biology and biophysics to try to design better malaria vaccines, in particular targeting the blood stage of malaria, which, as we'll see, is the stage of the malaria infection cycle when the diseases and symptoms of malaria are prevalent. So malaria is a huge problem up until this current day. It causes hundreds of thousands of deaths each year, and it causes hundreds of millions of cases. The vast majority of the deaths in malaria endemic regions are of children under the age of 5, because immunity to malaria develops, but this immunity is only partial, sufficient to prevent, by and large, the fatal symptoms, but still not enough to stop malaria from being a devastating disease. So while there are many different mechanisms or approaches that we can use to try to reduce malaria incidence, including the use of insecticide-treated bed nets, including drugs and medicines, the world urgently needs a malaria vaccine as well in order to try to stop and indeed hopefully to eradicate this disease. And I'm going to tell you today about one of the malaria parasites, the most deadly, Plasmodium falciparum. This is the life cycle of the malaria parasite, and as you can see, it's complex. It has a whole series of stages, both in a mosquito and also within an infected human. And the bit that we're interested in today is here on the right-hand side of this figure, when the malaria parasites burst out of the liver in this form called merozoites, and they infect and invade human erythrocytes. One malaria parasite goes into each erythrocyte, and some 10 to 20 parasites emerge 48 hours later. And so through this process of malaria, red blood cell invasion and growth within the red cell and bursting out again, the number of parasites in that infected person grows and develops, leading to the symptoms of malaria, many of which are due to anemia or due to these infected red cells sticking within the body. It's also at this stage that a small number of these parasites differentiate into a sexual form called a gametocyte, allowing the transmission of the parasite back to the mosquito. So that if we can stop red blood cell invasion by malaria parasites, we can stop the symptoms and the transmission of the disease. However, red blood cell invasion by the malaria parasite is a complex process for a number of reasons, and so it's been really challenging to develop a blood stage malaria vaccine. One of the reasons is that the structure of the cell that is involved in invading red blood cells. So you can see that here on the left. The malaria is caused by a single-celled eukaryotic pathogen, and it has these compartments or organelles up towards the apical tip, the point, this end of the parasite. And the machinery that the cell uses to drive its way inside a human red blood cell is held within these organelles, and it's only released from these organelles just when it's required. And as a result, this machinery is hidden away, protected from being detected by the immune system. So that's one of the reasons why blood stage malaria is hard to stop through vaccination, because of the protection of the machinery that's used for invasion within these organelles. A second reason why it's been hard to develop a malaria vaccine is illustrated on the right-hand side here. You can see the four main Plasmodium species which cause human malaria. And then in blue and red, you can see a set of different proteins which are used by these malaria parasites to make contacts between themselves and the red blood cell during the process of invasion. So these contacts are essential, these contacts are required for invasion. However, the parasite is very cunning and can use one of these molecules in the blue and red circles to make contacts with the red cell. But as the body learns to recognize that molecule and produce antibodies which block it, the malaria parasite can switch and use a different molecule to get in. So this redundancy, this ability to switch the machinery that it is using for invasion, is another reason why it's been challenging to work out which molecule to target through a malaria vaccine. And the last reason it's hard to develop a malaria vaccine against the blood stage of malaria is that it doesn't take very long for a malaria parasite to get inside a red blood cell. So it can only take, so from a malaria parasite bursting out of its previous red blood cell to the time that it bumps into another red blood cell and drives its way in, can only be, is often less than a minute, you know, with some 25 seconds or so from that contact between the parasite and the red blood cell to the point where the parasite is inside the red blood cell. So for all of these reasons, it's been hard to develop a blood stage malaria vaccine. We have a redundant system of invasion proteins, which can switch from one to another when one pathway is blocked. The process of invasion is fast, so there's not long for antibodies to act to block this process. And invasion proteins are held within these organelles. hidden away from immune detection until they're required, and their exposure is then transient. So as a result, we need high concentrations of antibodies present in our human blood, in our serum, to block this invasion process. So high concentrations of antibodies are required for protection. And so what we want to do is work out ways to not just get more antibodies, but to get more of the good antibodies, because the better the quality of those antibodies are, the more inhibitory they are, the fewer of them we will need to drive this invasion process. So developing a blood stage malaria vaccine has been difficult, but where are we getting to with it, and how are we using structural biology and biophysics to achieve that? Well, I'm going to tell you that there's an exciting candidate vaccine. I'm going to show you the structural and biophysical studies that we've been doing to understand how it works. I'm going to show you how we understand antibodies that block it. And finally, I'm going to tell you how we're using that information to conduct rational vaccine design. So first, of those different invasion proteins, and now I told you that this was a redundant system of proteins, but one of these proteins, called Rh5, was found to lie outside this redundant system, because Rh5 is essential for malaria parasites to get into red blood cells, at least for Plasmodium falciparum, the most deadly malaria of humans. So in 2011, Gavin Wright's group published that this Rh5 protein binds to a molecule called basogen, which is found on the human erythrocyte surface. I mean, in this graph, they demonstrated that MEM-M6, which is a basogen targeting antibody, could block red blood cell invasion by all tested malaria species, or falciparum strains tested. So in other words, this is not redundant, it's essential for invasion. And Simon Draper's group showed a few years later that if you immunize aotus monkeys in an aotus monkey model, and then you challenge them with malaria parasites, that those monkeys are protected. So on the y-axis here, you can see the fraction of these monkeys which did not require treatment with antimalarials because they did not develop parasitemia. And group B was one of the Rh5 vaccinated groups. And group A was the group that had not been vaccinated. So you could see that after 14 days, all of these animals developed malaria parasitemia to the point that they needed antimalarial treatment, whereas Rh5-based vaccination prevented that and was able to protect. Now, in early phase human trials, again by the Draper group, it hasn't been quite as effective as this. And so we don't want to use full-length Rh5 in our vaccines. We want to use Rh5-based immunogens, which just elicit the really high-quality antibody responses. So, firstly, how have we used structural biology and biophysics to understand the structure of Rh5 and its binding partners? Well, in 2014, we published our structure determined by Kate Wright in my laboratory of Rh5 in yellow bound to basogen in blue, demonstrating how this Rh5 molecule looks and how it binds to its receptor on the erythrocyte surface. More recently, we've demonstrated, in collaboration with Uwe Schultz, we've demonstrated, and this is work done by Abhishek Jamwal, we've demonstrated that basogen is not alone on the surface of erythrocytes, but instead it forms membrane protein complexes. Basogen here in blue is interacting either with a calcium transporter, PMCA, or with another transporter, MCT1. So Rh5 is not binding to basogen alone on the membrane, it's binding to basogen that's part of these membrane protein complexes. Next, Rh5 is not alone either. It forms a multi-component complex containing 4 other components. And Brendan Farrell, working in my group, has determined the cryo-electron microscopy-based structure of 3 of these components: Rh5 in yellow, CYRPA in blue, and RIPA in green, in this case bound to an antibody in light blue that targets CYRPA. And through a combination of cross-linking approaches and AlphaFold predictions, he was able to build a molecular model of all of these components. So yes, so this is just to show that through cross-linking mass spec, using cross-linking and then mass spectrometry to find out which residues are close to each other in solution, in 3 dimensions in the protein structure, Brendan was able to piece together the structure of this protein complex. And in particular, he was able to determine the first structure of RIPA at molecular level of detail. And RIPA is a large complex molecule formed of these multiple domains. And actually, Brendan was only able to see the RIPA core, as we've called it, and not this region that follows the RIPA core, which we call the RIPA tail. And through AlphaFold predictions, this is what the RIPA tail looks like here on the right. We believe that it's an elongated, extended structure which emerges from the RIPA core, protruding from this region of electron density. So by combining all of these pieces of structural and biophysical information, we've been able to build this model for how this whole protein looks. And then Brendan was also able to show that, I told you that this was a 5-component complex, but I've only shown you the structure of 3 of these components. The other 2 components, CSS and PTRAMP, form a complex which is attached to the parasite membrane. And Brendan was able to show that full-length RIPA and also just that RIPA tail are both able to bind to CSS and PTRAMP with the same affinity. And he did this using surface plasmon resonance measurements. So to summarize all of that data, we know that basogen is present on these membrane protein complexes on the erythrocyte membrane. And we know that PTRAMP and CSS are found on the merozoite membrane. We know that RH5 is part of this 3-component complex, which we now see the structure of through cryo-EM and AlphaFold predictions. And we know that this PTRAMP and CSS complex is on the parasite membrane, in which it interacts with the RIPA tail. So in other words, this big protein complex spans the parasite membrane all the way through to the erythrocyte membrane, generating this bridge which links these 2 membrane protein complexes together. So we're starting to understand through structural biology how parasite and red blood cell are linked during these essential invasion processes. So can we use that information to design better malaria vaccines? And this uses the classic approach of structural vaccinology. And this is all done in collaboration with Simon Draper here in Oxford. So Simon's team vaccinates human volunteers with RH5-based vaccines and then use single B-cell sorting to get antibodies from these and then assess the quality of these antibodies. My lab understand where these antibodies bind across the protein components that they target and use that to try to design more focused vaccine immunogens. So how do the most effective RH5 targeting antibodies work? So we're studying all 5 of these components, but I'm just going to tell you about RH5 today. So human volunteers were vaccinated with RH5, single B cells were sorted, and antibodies were cloned and tested. And this was done by Dan Alanine, a joint student between Simon's lab and my lab. And Dan was able to group these antibodies into classes. So within each of these rectangles, you can see a set of antibodies which can't bind to the antigen RH5 at the same time. In other words, these are called competition groups. And on the y-axis, you can see the EC30 of the growth inhibitory activity. So low concentrations of antibody are able to block growth or invasion of red blood cells by malaria parasites in the red and the blue cases. And then we were able to study these different antibodies and understand how they bind and where they bind. So first of all, again, using biophysical techniques, surface plasmon resonance, Dan looked at the EC30 of these different antibodies, and he looked at the KD, he looked at the off rate, and he looked at the on rate that these different antibodies had for RH5. And the best correlation between what these 3 different kinetic parameters of binding and protection or growth inhibition was actually the on rate. So in other words, antibodies that bind fast are better in malaria. And if you remember, I told you that it only takes seconds for a malaria parasite to get inside a red blood cell. And that's why these antibodies only have a brief window at which they can act. So antibodies that bind quickly are better than antibodies that bind slowly. Dan was also able to use crystallography in this case to understand how the red antibodies and the blue antibodies binds to RH5 and to understand where their epitopes are. So we know where within the RH5 molecule the top quality antibodies bind and how they bind. And we also know through comparison with Abhishek's data on these membrane protein complexes that these best quality antibodies, for example, the red one and the green one here, project off RH5. So these red and green antibodies are binding to RH5, but they will be preventing RH5 from binding to Basigen because in that conformation, they block the interaction with the red blood cell membrane. So these antibodies bind to that top half of RH5 as we imagine it, away from the CYRPA binding site, where they will clash with the membrane, stopping RH5 from binding to Basigen. So lastly, just for the last couple of minutes, how can we use this information to design improved vaccine candidates? Well, we now have a load of different structures of antibodies bound to RH5. You can just about see RH5 in the middle in yellow, bound to all of these different antibodies in different colors. And all of the growth inhibitory activity positive antibodies, in other words, the antibodies that can stop malaria parasites from getting into red blood cells, these all bind to the top half of RH5 above that dotted line. And the best antibodies are the orange and red ones here, which approach this particular surface of RH5. And the less effective antibodies bind down at the bottom. So we're in the process of designing and producing a set of vaccine immunogens to test these, in which we focus down on the key antibody epitopes. So RH5.1 is full-length RH5, which has already been tested in human clinical trials by Simon's group. I'm going to tell you in the next couple of slides about this antigen here, which is a large part of RH5, but it's been truncated to remove large flexible regions at the N-terminus and in this central loop. And it's also been thermally stabilized through methods that I'm going to tell you about in a minute. And this vaccine immunogen is under clinical assessment at the moment. We're also designing vaccine immunogens using protein design methods, which contain just the top half of RH5, where all of those good antibodies bind. And lastly, this immunogen where we've taken a synthetic scaffold and we've grafted on the key antibody epitopes for those orange and red antibodies. So I don't have time to tell you about these today, but this is the most progressed. I'm just going to give you a couple of slides on that before I finish. So this was a collaboration with Sarah Fleischmann and A.D. Goldenzweig at the Weizmann Institute, who developed this really beautiful method whereby you can use Rosetta-based protein design to repack the core of your protein to make it more thermally stable and to make it fold better. The reason we did this was because RH5 is quite hard to produce, and a vaccine for countries which don't have a high growth domestic product, one would wish the vaccine to be cheap to produce and to be thermally stable. So this method works by doing a multiple sequence alignment and looks for variable sites across that sequence alignment. And then it allows those sites to vary in silico, and it uses the Rosetta scoring function to score whether those changes are likely to make the protein core more stable and the protein fold better. And so we built up a set of somewhere in the region of 20 mutations, which collectively made the protein more stable. And indeed, this worked. On the gel on the top left, you can see the production of RH5 in E. coli. You can see no protein is produced at all, whereas the HS1, 2, and 3 variants, these thermally stabilized variants, expressed really well in E. coli. You can see that it binds to Basigen with the same affinity. The structure was identical, but the thermal stability was far stronger. And so this vaccine immunogen, which is focused because we remove these flexible regions, which are the sites of binding of non-neutralizing antibodies, and which is thermally stabilized, is now in production at the Serum Institute of India and will be used in a phase 1 clinical trial later this year. as we continue with our previous vaccine development strategies. So our conclusions from this talk: the structure of this RCR complex in RH5 reveals this bridge which links the parasite and erythrocyte membranes, and is every component in that bridge is a vaccine candidate and is essential. Secondly, we've used structural studies of the best antibodies to find how antibodies neutralize invasion and how they should be presented in a vaccine. And finally, we're using structure-based design to generate improved vaccine immunogens, which elicit just the good antibodies, the higher quality antibodies. And the first of these is in production for a phase 1 clinical trial at the moment, with other ones in preclinical assessment. Now, as I went through, I think I mentioned the people involved in this work, but here are some of their faces, and we're very grateful to the Medical Research Council, USAID, and the Wellcome Trust for funding. And if you're interested in our work, do check out our website there as well, higginslab.web at Oxford. And thank you all very much for listening. I will unshare because we're on a tight schedule and hand over to the next speaker.
Thank you very much, Matthew. And we will switch over to our next speaker, Professor Bimshai Shayan, who will talk about, okay, here you go. So please, Bimshai.
Okay, all right. Okay, well, first of all, I would like to thank all the organizers for inviting me to participate in this wonderful event. And so I'm speaking, from Thailand, and this is the picture of our institute, which is in short is a long name, but you can call our institute and in short as VISTEC. And so we are in Rayong, which is in the green area, and in the innovation park that the Thai government designed that this would be another hub for the innovation in Thailand, about 2 hours from Bangkok, about 1.5 hours from the major airport. And so my in the lab, that my fundamental part on the basic science stem from our studies in the various reaction of redox enzyme in enzyme mechanisms. And we have been exploring in the various systems, particularly on the flavin-dependent enzymes, mechanisms, and catalysis. And in recent years, we have expanded into the enzyme engineering, biocatalysis, and as well as in the synthetic biology, metabolic engineering. And also we try to apply our knowledge whenever that it fits into the detection and the bioreporter systems. And towards the SDG goal that our group have been collaborating and working on the various systems to support circular economy. So in the way that we see this as an important tools for the sustainable future, because we generate a lot of waste, and particularly the bio waste can be converted by various enzymatic system and or the metabolic engineer cell to make valuable compounds from the low value waste or the biomass. And our lab, our goal is also work towards several projects to support the sustainable development goals, with the Thanyapon Wongnet collaboration in the broader network group and in the various groups as well. We work on, not only on the enzyme and metabolic engineering, but as well as the anaerobic digestion and process engineering. So with this, we also work with the community that where we selected the starter for efficient the anaerobic digestion and put this into the zero waste digestion. So then this way it promote people to segregate trash. And so other type of the dry trash can be used in the recycled manner for the organic waste or food waste. Then it can be used by this anaerobe to produce biofertilizer and biogas. But for the story, the major story that I'm going to share with you today is on the enzymatic system that can help on the pesticides decontamination and on the pesticide detection. So pesticides decontamination and pesticides poisoning is a big problem worldwide. particularly in developing countries such as in Thailand. So all around the world, is like 100 million cases have been reported to be having problem with pesticides poisonings, and majority of these victims are poor farmers. And among pesticides, organophosphate and organochlorine are also major groups of pesticides. So with this, once it get into the environment, it get degraded, particularly by esterase or etherase, and to generate the core structure, which is a phenolic derivatives. This type of the phenolic derivative is still toxic, to human or to animals, because it contains phenolic derivative, contain halogenated phenol or nitro, containing group of phenol. So we have been studying from basic science part, we have been studying one particular enzyme, we call it HAT-A, monooxygenase, by solving the structure, three-dimensional structure of these enzymes and study reaction mechanism. And we found that, understand that how it's work by the enzymes bind to the, let me just put my pointer. So by binding to the, this structure, it's a structure of the FAD. So the enzyme using the reduced flavin at the starting, the starting active complex. Reduced flavin reacting with oxygen to form a reactive species called C4A hydroperoxyflavin, which can incorporate this terminal hydroxyl group into the phenolic substrate. So with this, for the compound to allow the compound to eliminate the toxic halide group and to generate benzoquinone, which is it can be reduced and re-aromatized to be the phenolic product. So we have been working for this enzymes for, as you can see in publication list, for more than 5 years, about 7 or 8 years. We understand the system, in the basic science, completely. And so one of the issue that amazed us for quite some time is that this enzyme is good for, can use a wide variety of phenolic derivatives as substrates. So we've been wondering what can be the application on this part. And in 2016, it was lucky that I attended the conference in Japan, so that on the bioluminescence conference. So then I listened to one of the talk by Professor Oba from Japan. So he been sharing the discovery that benzoquinone, once is condensed with a D-cysteine, and then it can form D-luciferin, which is the substrate for the enzyme luciferase, particularly a firefly luciferase in this structure. So then we got the idea that because we have the enzymes, which is like HAT-A, that can detoxify or eliminate either the halide group or the nitro group from phenolic substrate. So that can be generated to form benzoquinone. So we got the idea that how about we couple the two reactions together? So if we provide a system with the D-cysteine, now we can convert the phenolic compound into the valuable D-luciferin. So we tested the idea, and if we found that indeed the couple reaction can generate the D-luciferin with the properties exactly the same as the synthesized luciferin. So it can also generate light that can also, to emit light by adding the firefly luciferase into the system. So then with this concept, that was we published this work 3 years ago. So it's a concept that we can couple enzymatic reactions, that we understand it can use variety of the phenolic derivative to do 3 things. So the first thing is that to use the enzyme to do the detoxification of these halogenated compounds, and then to synthesize valuable compound like luciferin. But when we add the luciferase, now we can generate the application or innovations of the detections of pesticides. And then, but by that time, 3 years ago, we know that we published the work, but we know that we need to improve the work further. because the yield of the luciferin synthesis was quite low, and it could only, this couple reaction could only use phenolic compounds, but not the real pesticides. So we further developed. So then this is using the program, particularly on the FirePlot program, to generate candidates of the mutant to make the enzyme more stable. So then we can increase thermal stability of HAT-A. So then this way HAT-A can be used as a toolbox. So we further develop the cascade reaction to generate the continuous supply of the cofactor. So then we can generate the D-luciferin by converting the phenolic derivative. This way, we could improve the yield up to 50%. So now we have a toolbox enzymatic cascade because it's HAT-A for preparation luciferin. We call the reaction called HELP. So with this reaction, now we tested reaction with various the phenolic substrate. So in overall, we tested 17 phenolic substrate. We found that this reaction can convert variety of these phenolic compounds, which is derived from either from agricultural like usage from the pesticides, and it can also be generated by industries or by the environmental use of this phenolic derivative. So with this reaction, it can convert this phenolic derivative 17 compounds into 8 types of the luciferin. And to our we were glad that we could also synthesize the new compounds, new luciferin as well. So with the 4,5 dimethyl luciferin and 5 methyl luciferins, these new compounds in fact generate light in the longer wavelength, such as generate the color almost in the red color. So this can be quite useful for the applications for people, particularly using this luciferase in animal applications. So this way, this luciferin can also generate the brighter light than the native luciferin. So currently, we are developing 3 application based on these systems. So the first is to use a HAT-A to do the bio detoxification. So then this way we hope that to come up with a solution to do the decontamination of the pesticide detection, and the ability of the HELP reaction to synthesize variety of the D-luciferin derivatives. We hope that in the future can contribute to the many colleagues that will work with the animal testing system. So then to make the system more efficient on the monitoring the bio reporter on site without with the reduce the number of the animal requires. And for the third application is to use the system to do the detection of the pesticides decontamination. So with this, we developed the technology we call LUMOS. So LUMOS, just like in Harry Potter, where you have light, where we have pesticides now, we should be able to see light. So with the luminescence measurement of the organophosphate derivative, so we constructed the reaction now with the real starting precursor as a pesticide, such as parathion, EPN, profenophos, and the various compounds, and we use the etherase and the SRS to generate the first to degrade into a phenolic derivative, and then later on couple it to synthesize luciferin. So once we put this system, and then we tested the system ability to detect these pesticides. So the good part about having the signal of the luminescence is that we don't need to pre-treat the sample. Even the dirty sample, the crowded sample, such as in the urine, synthetic urine or serum, or the solution that we wash from fruits from the environment. With this LUMOS technology, the detection level is actually can go down to the part per trillion. So it's equivalent to the sensitivity of the LCMS method. And so we tested ability of these compounds. As you can see that this ability, actually, we can even visualize this by eyes, as I will show you in this video. So the whole work, if you are more interested in this work that we also just published this last year in Angkawante Chemie, and this expressed a lot of the interest from many international news outlets. Just last week, I just came back from the province in the northern part of Thailand, a province called Nan, where we have the project together with the social scientists that we work and try to develop and implement our technology for the real use of local people. So in Nan province, there are a lot of this contract farming where there are a lot of usage in the pesticides and a lot of this chemical fertilizer in growing maize that to make it for animal feed. So with Lumos technology, we help the farmer in the village that in called Prong Sanuk that they want to monitor the system, the contamination of these pesticides in the water, in the soil. So we collected the sample, and with the technology, it's simple enough to work on site in the province where you don't require sophisticated laboratory. So together with the anaerobic digestion team, we work this as a model that hopefully that we use the Lumos technology to detect, to increase awareness of the local people in terms of they need to monitor the level of the pesticide contamination. But with the zero waste technology to use the food waste to produce the biofertilizer. So then we provide them with tools in order to do the more of the healthier and safer the agriculture. So then this way they promote them to grow the high value herb instead of the growing the high utilization of the pesticides. So this work have been involve a lot of people in a big team, particularly the one on the left, the Prachaya Wetaisong, which is the whole Lumos project was his PhD thesis. But now, he's graduated and then trying to further develop the technology that, try to work on this to implement this to be in the real usage. And also various people in the team that have been contributed to this technology all along, and have to thank all the people who work together, not only my lab, but in many labs that we work together and as a network. And thanks to all our funding agency that to support the work in our group.
Thank you very much, Pimshai. This is really exciting science. And thank you also for sticking to time. So we are now moving on to our next speaker, Jesus Perez Gil from Madrid, who will share his screen.
Good morning from Madrid. Good morning, everyone. I hope you can hear me well. Let me see.
Yes, very good for the sound.
Yep. That should be my screen.
Excellent.
Yes, perfect.
Great. Thank you very much. First, let me tell you that it's a pleasure for me to be here and an honor to share with you a bit of the research we have been making for many years, trying to apply biophysics for designing better respiratory medicines. First, let me introduce what is the reason why we are coming to biophysics in connection with respiratory mechanics. What is important for you to realize is that in our lungs, we are exposing about 100 times more surface to the environment that we expose in our skin. That is a challenge from a physical point of view because we need to maintain such a large surface fully exposed to air continuously, while we protect it and defend it against the potential entrance of pathogens. But there is another challenge because this surface is very dynamic. So for you to realize, we are introducing in our lungs about 15,000 liters of air per day under resting conditions that can go up to 50,000 liters of air per day. And within this air, we are introducing contaminants, particles, potential pathogens, and very close to the blood, where we need to expose this air in order to get the oxygen we need in our metabolism. So as I said, this is a major problem from a physical and a biological point of view. The system we have been working during the last 20 years or so is related with the defense and the stabilization of such a large surface. If we go to the real structure of a lung, here you can have an electron microscopy picture of a lung from a mice. You go to some detail, you see the structure of the alveolar spaces in the lung, where the surface is exposed to air and the air and oxygen exchange occurs. But what is important to realize is that this surface is wet. So there is this yellow line is indicating that there is a very thin layer of water covering the surface of our lungs. This is important because within this water there will be some important molecular mechanisms to maintain an innate defense system in charge of protecting our lungs from the potential entry of pathogens. But the water is a physical problem because within this bulk aqueous phase, the water molecules establish polar interactions among each other, but the water molecules that are at the surface don't get enough molecules to compensate those interactions. The result is that these surface water molecules have a net force component, which is called surface tension, which means that opening this surface costs energy. And that energy is against the increase in surface we have to do in each of our breathing cycle. When we inspire, we open our lungs. More water molecules need to go up to the surface, and that costs energy because they need to break interactions that cannot be formed at that surface. And the pressure that is opposing to this opening is defined by the Laplace law, which means that the pressure that is pushing to close these air chambers, like the alveoli, is directly proportional to the surface tension, gamma, and inversely proportional to the radius, meaning that the smaller the alveoli, the stronger the pressure to be closed. This is a major problem. The evolution has solved that by creating a system which is a pulmonary surfactant, which is a surface active agent, which is synthesized and secreted to the surface of our lungs. There it somehow substitutes water molecules from the exposure to air, and that reduces surface tension and therefore the force opposing to opening our lungs. It's important to realize that in the absence of any surface active material, we will need more than 60% of the metabolic energy used for the mechanical work of breathing, but in the presence of surfactant, we need only 3% of that energy. And that is the importance of such a system. And the system, and here you can have a scheme showing that in our alveoli, we have specialized cells, 82 cells, alveolar type 2 pneumocytes, in charge of synthesizing and secreting this material, which is rapidly absorbing to the air-water interface, reducing surface tension and facilitating breathing mechanics. Well, if we go to some details, we will see that this is the way that the alveolar epithelium is continuously stabilizing the lung. And you can have here a picture in which you see how the pneumocytes are synthesizing this material. Here it is stained with laudanum, which is a lipophilic fluorescent probe. Each of these organelles is a lamellar body. Here you have some detail in an electron microscopy showing how surfactant is stored in the form of tightly packed dense membranes, which upon secretion absorb to the air-water interface, form a multi-layer film. This is excluding the exposure of water to air, therefore reducing surface tension and facilitating stabilization of such large surface. Some elements in this material are also intercalated in these layers and participating in innate defense, so that at the same time, this material is stabilizing physically the alveolar water interface, but also defending biologically our organisms from the potential entrance of pathogens. Well, there's different pathologies related with the lack or the dysfunction of pulmonary surfactant. One of the better known pathologies is what happened in the immature lungs of preterm babies when they are born before their lungs are maturated and have synthesized the surfactant that is required for establishing the air respiration. If this is the case, the lungs cannot be open of these premature babies because they don't have the material required to stabilize the air-water interface. Now we can solve these problems because these preterm babies are treated as early as possible after their delivery with an exogenous material to replace what the lungs have not been able to synthesize yet. And if you look at the mortality of premature babies along the last year, you see how late in the late 90s, in the last century, there is a very sharp decay in mortality as a consequence of the establishment of this pulmonary surfactant exogenous therapy. I have taken some data. The last data are from 21, showing the difference in mortality before and after the establishment of this pathology, but also to show how there is still a long way to improve the situation for the mortality of these preterm babies in different countries. And this is something that depends not only on the, let's say, spreading of this technology on supplementation with an exogenous surfactant, but also the critical care of babies that are born very early and they require very extreme critical cares. Well, the research in this field are dedicated mainly to understand the molecular mechanisms of occurring in surfactant and using this knowledge to produce better therapeutic materials. And we have been collaborating, for instance, with different pharmaceutical companies to produce new surfactants in which we can combine lipids and synthetic proteins and peptides to make a material that can be used extensively for the treatment of different pathologies. We need other materials because there are other problems related with inactivation of surfactant when there is lung injury and inflammation in the lung. That creates an environment in which phospholipases are liberated, oxidation as a consequence of inflammation is inactivating surfactant, and again, we need to replace that material for the lungs to work properly. This is also what is happening, for instance, in lung injury as a consequence of COVID-19. And we are now developing studies to understand how we can restore surfactant function in these cases of a lung injury, but that requires development of new surfactants that are resistant to inactivation in enough amount to be given in a repetitive way to the patients. How we can study that? Well, we have developed over the years some in vitro techniques to mimic the mechanics of lungs in let's say, simplified systems. This is what we call a captive bubble surfactometer, in which we can measure the action of a pulmonary surfactant preparation against an air bubble. This is an air bubble in this video that is somehow mimicking how an alveoli is working. Here you can see how the injection of a very tiny amount of surfactant produces, upon absorption to the surface, a change in the shape of the bubble. This change in the shape is a consequence of a reduction in surface tension. So we can monitorize from these kind of videos, we can monitorize the change in surface tension and measure surface tension against time. And here in this plot, you can see how injection of surfactant, which is working properly, is reducing surface tension in a fraction of a second from 70 millinewtons per meter, which is the typical surface tension of water, to less than almost 20 milliliters per meter. Then we can compress and expand the bubble using a piston, and it is represented here. And in this video, you can see how the shape of the bubble is changing further upon compression and expansion, mimicking compression, expansion dynamics during breathing. We can represent surface tension against area, and you can see here how this surface tension upon compression is reduced even further to less than 2 millimeters per meter. This is what is required to stabilize the lungs. And it does this in a very reproducible way upon many cycles, which is what you need for stabilizing your lungs. This depends on a proper composition and structure of the material. If the material is not working properly because the composition is, for instance, depleted of proteins, then it doesn't work as stable as it should. We can use this to monitorize how surfactant is altered in different pathologies by taking, for instance, material from patients. Here, we can compare, for instance, this behavior of surface tension against area in material taken from babies with no lung disease, and compare that when we use material taken from the lungs of babies that suffer of meconium aspiration syndrome, which is a pathology as a consequence of accidental inspiration of meconium during birth. That creates lung injury, inflammation, and inactivation of surfactant. And you can see how in those babies, the material never reached low enough surface tension, indicating that the inactivation of surfactant is one of the factors inducing this pathology, and that could be treated as it is in these babies by replacement with exogenous surfactant. And in the last few minutes, let me show you that the research in this pulmonary surfactant biophysics is coming to a new line of application, because as I said, the lung surfactant film that is coating the alveolar spaces is the first material that is encountered by any kind of molecule or entity which is inhaled into our lungs. That, of course, is important in order to understand toxicity of inhaled products, for instance, pollutants or contaminant particles or any kind of toxic material, which effect on surfactant can be part of the toxicity. But it's also important if we want to design much better inhaled medicines. For instance, if we want to deliver drugs upon inhalation, we need to understand how the drugs interact with surfactant, because depending how they will interact, they will end deep into the lung or they will be cleared and removed away from the airways. And we can see here, for instance, how the delivery into the trachea in an animal which is monitored, for instance, in this case, a mice, is monitored by infrared fluorescent tomography. Here you see how the injection of a material in the trachea monitors over time, doesn't move very much, is not spreading well around the airways. But if it is combined with the pulmonary surfactant with a proper composition containing the lipids and surfactant proteins, then very early upon delivery in the trachea, the material is detected in the deep lung, and taking the lungs out, one can see how the material is completely homogeneously distributed, meaning that surfactant can be very useful as a drug delivery agent upon combination properly with a drug. And we have developed in our laboratories in vitro systems to monitorize this kind of spreading promoted by surfactant. For instance, a device like this in which you can combine donor and receptor compartments connected by interfacial bridges, somehow mimicking what happened when you deliver something in the upper airways upon inhalation, and through the conductive airways, they have to reach the deep alveoli for that to have a proper effect. We have developed surface balances in which we can monitorize how material is moving from donor to recipient compartments using the interface as a pathway. And we have seen how the proper combination of a drug, like for instance, a corticosteroid like beclomethasone with surfactant, is taking the material in a very efficient way from the donor to the recipient trough. If you combine the same drug with lipids, only in liposomes, which is a classical way to deliver drugs that are not properly soluble to make them more bioavailable. Then they never, in that case, only in liposomes, the drug never moves away from the donor to reach the recipient trough. And we have seen, and here you can see how different pulmonary surfactants, including material we can purify from lungs or the clinical surfactants that are being used to treat babies, they are both very efficient to promote delivery of drugs. And we have seen that also in vivo in collaboration with the group of Jahar Bhattacharya in Columbia University, using a mouse in which lung injury is induced by instillation of lipopolysaccharides, endotoxin, and treating those mouse intranasally, either with an anti-inflammatory drug, tacrolimus, or with surfactant, or the combination of both. You can see here how lung injury is seen by the presence of blood in the airway of the lungs, and you see that as a consequence of inflammation and edema. This edema is not prevented at all if the mouse are treated only with the anti-inflammatory drug or only with surfactant, but is much more prevented upon treatment of the mouse with a combination of surfactant and the anti-inflammatory drug. And we see that also by checking in the infiltration of white blood cells as a consequence of inflammation, which is very much promoted when the drug is properly combined with surfactant. And in that case, we can see how the drug ends in the macrophages, which are the immune cells that are somehow protecting the lung deeply in the alveolar spaces, meaning that the drug is arriving that massively and facilitating also the transepithelial delivery into the blood and the systemic spreading. So in our more promising research lines, we are now developing different strategies to use surfactant as a drug delivery agent, in which we can facilitate not only the delivery of drugs, but also nanoparticles, which can carry nucleic acids, for instance, to facilitate gene therapy or gene silencing upon inhalation delivery, which is a very efficient way to take it in this case in a let's say patient-friendly way. So with all this, I hope I have been able to convince you that biophysics and the development of proper in vitro models can facilitate the development of new strategies and new therapeutic products, and that this can be done in a way that can be easily extended to all countries and then facilitate a much better health treatment for everybody in the world. And here, let me just finish my talk by showing you our group, which is a combination of physicists, biochemists, physiologists, pharmaceutic, which is a kind of multidisciplinary work, which is required for this kind of approaches. This building you have at the bottom is the biology faculty here at Complutense University in Madrid. And let me also thank all the funding agencies that have facilitated this over so many years. And thank you to all of you for your attention.
Thank you very much. This was really enlightening. So we will now move to our next speaker, Miguel Gestano. Do you manage to share your screen?
Yes, first of all, well, good morning or good afternoon or good night, wherever you are. Let me just then start here my sharing. I will perform something. I will perform a presentation that is more devoted to, well, let me see, can start here. Are you seeing my presentation now?
Yes, but it's still not full screen.
Now it's okay. So my presentation will be more like a communication exercise than exactly tell you about the details of my science. I'll try to highlight how our biophysics is important, for instance, to try to be prepared and ready for the next big pandemic, which is now a big concern. Okay, so we went through the COVID pandemic, and we know that future pandemics will come. We want to be prepared, we want to be ready, which is kind of a dilemma, because one of the main characteristics of a pandemic is that you cannot exactly predict when the pandemic will come, what will be the agent that will cause the pandemic. So how can we be prepared for something that we don't know what it is exactly, and we don't know exactly when it comes? So, and how can we think about how to actually control the impact of the next big pandemic? Moreover, what was the role of biophysics in actually controlling the impact and saving lives during the COVID pandemic? Here we have a timeline and several pandemics, mainly the Spanish flu here, the AIDS pandemic here. The dimension of these fuzzy balls is actually corresponds to the number of victims of dead people during those pandemics. We during the COVID had 7 million reported victims, mortal victims. The WHO estimates that the real number can be as 3 times higher than that. But still, when compared to the, let me just, okay, let me just bring my pointer here. Okay, so when comparing the dimension, okay, of the number of cases of the COVID-19 with AIDS or the Spanish flu, we see that it's relatively lower for a population in the world that is much higher. So somehow we were able to contain, okay, the number of cases and the number of deadly victims in this pandemic, and we are actually trying to achieve more and do better the next time. Now, pandemics, or the potential from pandemics, is higher than we tend to think. It's higher than we usually think of, because the number of pandemics actually it's relatively infrequent, but the outbreaks of potential pandemics are relatively frequent. This is also a timeline for the 21st century, so it's only after year 2000, and there are several outbreaks with pandemic potential that occurred, okay, and they are very frequent. And we know that the dynamics of viruses in nature, they include several species, they can adapt to several species, and they are actually subject to all kind of unbalances that the environment is facing now. So we have to understand the basic biology of the viruses to actually be ready to understand how they will evolve, and so what is the potential for a certain pandemic in the future. So my point is that you really to understand the biology of the virus first, so that you can control the disease in the end, and that's where biophysics comes. Of course, one of the ways that you have to control the future pandemic is to develop broad-spectrum antiviral drugs, okay? Drugs that are potentially able to inactivate a very broad range of different viruses, so that potentially it can include any virus that will appear in the future. Okay? And so even the World Health Organization is actually trying to devise politics and measures and incentives to be prepared and ready for the future of pandemics and epidemics. And our research, which is basically biophysics research, follows this idea that we have to be prepared and that we have to be ready. And so how can we use biophysics, okay, towards this goal, to protect humanity from the next big pandemic? Well, we're trying to focus on one family of virus that is actually carried and distributed by a single species or mosquito, or a single genus of mosquitoes. So they are very well adapted to these mosquitoes from the genus Aedes. There are two main species in this genus, but the virus such as Zika, dengue, yellow fever, chikungunya, they are very well adapted to these mosquitoes, and they are very well adapted to humans too. Because now these mosquitoes will sting or bite, okay, humans, and so they actually distribute all these different viruses to humans. Okay? So we thought this would be a good family of virus to test a broad spectrum antiviral, because they are different viruses among themselves, but they are all adapted to humans, and they were all optimized in nature for two species, mosquitoes and humans. And mosquitoes and humans, of course, are very different. Moreover, this problem is big. Okay, it's huge now in the globe. But with climate change and global warming, the areas that these mosquitoes actually colonize is spreading. So the threat, which is already huge, okay, is getting huger. So we thought we would concentrate on these viruses, try to develop a broad spectrum antiviral using biophysics, and that the lessons learned could be actually used eventually to create even broader spectrum antiviral drugs. And so far, there are no medicines available. So when you cannot actually tackle the problem of the virus, then you go after the mosquitoes. And for instance, in all South America, you can find warnings like this, so that you control the reservoirs of still water, so that you can eventually control the population of mosquitoes, and so try to control the problem from the side of the mosquitoes, because you don't have drugs to antiviral drugs. You have drugs to control the effects of the virus, but you don't have drugs to actually inactivate the virus itself. So using biophysics, we had to imagine what could be, okay, the target in the virus that could be used and would grant a broad spectrum antiviral activity, because you have to have a common feature among all these different viruses, so that you can actually attack the viruses using this common feature, this common target. And if we look at the structure of dengue, Zika, even Ebola or HIV, which are not in this class that is carried by these mosquitoes, the common feature is that they all have an envelope, they all have a membrane, they all have a lipid bilayer. And now we are thinking molecularly, meaning that we are thinking in terms of biophysics, okay? Trying to devise a drug is an exercise that requires molecular thinking. So, and if you think the interaction of molecules, you're thinking biophysics, okay? That's the point. So we tried to develop a drug that could actually attack the membrane, the envelope of the viruses. At variance with cells, viruses cannot repair damage on the membrane. Okay, cells can, okay, but viruses cannot. So a mild lesion for a cell would be a devastating lesion for a virus. So that's the initial concept, and we started this project that is funded by the European Union called No Viruses to the Brain. I will not go into details, but if you want to know more about this project, it's here in this website. And the most challenging of all the viruses that we are trying to address in this project is actually Zika, the Zika virus, because it is able, the virus, to cross the blood-brain barrier of an adult, that is to say, to cross the arteria in the brain. So it goes from the blood to the brain, and it may cause, it doesn't happen to all but it may cause to some neurological lesions. So lesions in the brain have effect in the brain. But dramatically, it can also cross the blood placental barrier, reach the fetus in pregnant women, and cause severe neurological damage to the fetus, to the baby that is developing. And this is devastating. This is one devastating feature of Zika virus. So we used Zika virus as our test case, okay? And because it's also the most challenging. We have to find a drug that is able to cross the blood-brain barrier, to cross from the blood to the brain and protect the brain, to cross from the blood placental barrier, that is to reach the fetus and protect the fetus. So that's the ambition, challenging from the beginning. We knew from previous work using biophysics, using our biophysical reasoning, that a certain class of molecules, that is porphyrins, they are able to insert in the envelope of different viruses, okay, and produce lesions in this envelope. So we had a basic structure for a broad antiviral molecule. But we need to place it in the brain. We need to place it in the fetus in the case of pregnant women. And of course, we need to test better the efficacy of this molecule in inactivating viruses by destroying their envelope in vivo. That is to say, in an animal setting, in an animal environment. Okay, because in a test tube, it's more or less easy. But in a physiological environment, inside an animal, inside a person, it's different. So we had this molecule that could potentially fit the purpose, but we needed to transport it to the brain. What was the solution then? Well, we used actually parts of proteins, domains of proteins from dengue virus, ironically, from the capsid of the dengue virus. So we went to virus to seek for tools that would enable us to fight the virus, which is kind of ironic for the virus. But viruses are actually molecular machines that were optimized by nature to enter cells. So it's not at all surprising that we go and look for tools to transverse cells in virus. So we found these called peptides, okay, these domains of proteins in viruses that could help us actually take the antiviral molecule inside cells and across cells. So that could help us carry these antiviral molecules from the blood to the brain or from the blood to the placenta. Okay? So what we did in the project was simply binding these porphyrins, these antiviral molecules, with these peptides, with these fragments of proteins into a conjugate. And we hoped that this conjugate would maintain the characteristics of the antiviral molecule and the transport molecule. Okay, so we would hope that this construct would cross from the blood to the brain and it would inactivate viruses there. So we started by synthesizing these molecules, of course, and then again comes the molecular reasoning. You have to know the molecules, you have to know the specific groups, chemical groups of the molecules, so that you know how to combine them. This is a purely molecular science work. Okay? And then we tested the molecules for their capacity to translocate across the cells that constitute the walls of the arteria in the brain. We did that in vitro, so we grow the cells to separate the compartments, and then we test the transport across the compartments, and We use cells for the blood-brain barrier. We use cells for the blood-placental barrier, and we test and compare to a control, of course. We test the capacity of these molecules to go from one chamber to the other. Then for antiviral activity, we started checking the antiviral activity with HIV, not with Zika initially. We started with HIV because it was easier, basically, because it's from the AIDS pandemic, we have a lot of knowledge and a lot of capacity to test anti-HIV activity. And HIV also has an envelope. So in principle, if it would work for all viruses that have envelope, it would also work for HIV. And the first test was for HIV. And we found that at relatively low concentrations, actually we get antiviral activity from the molecules. So from the first experiments, all in vitro, meaning all in the laboratory, we knew that there was the potential to cross from the blood to the brain or the placenta, and we knew that the antiviral activity was maintained. Okay. Then we tested different conjugates here in different colors for Zika and dengue, for antiviral activity in Zika and dengue. This is kind of the number of virus present in a sample. This is concentration of the drug, okay, of the tentative drug. And you'll see here that in some cases, some conjugates are actually able to cause severe drops, okay? These are orders of magnitude, severe drops in the quantity of viruses. So they are actually also able not only to control the replication of HIV, but also Zika and dengue. Not all conjugates work for all the viruses, which challenges the idea of a broad spectrum antivirals from the beginning, but that is science. I mean, it doesn't come as, the results are not always what you have planned for and resulted. So the important here is that some conjugates are actually having the activity we hoped for. Then the final the tests were for the way that the molecules use to cross from the blood to the brain, and there are different ways to go across cells from one side to the other of epithelia, and then scientifically and with a biophysics reasoning, we selectively inhibit some, okay, some steps here. We then check the result, and so we try to reconstitute what is the pathway of the molecules in the cells of the arteria so that they can cross from the blood to the brain or to the placenta. These are the results. So there are actually two inhibitors that actually have an impact on the crossing. So we know that the molecules use certain routes, specific routes, and what these routes are across the cells. This is important to develop other molecules in the future. Then we perform also some animal tests, and the animal tests are challenging here because, for instance, Zika or dengue, they are human virus. They are not adapted to rodents, for instance. So you don't have Zika disease in mice, for instance. So it's difficult to test the drugs in mice because they don't get Zika disease. We have to create a situation which is Zika disease-like in some animals, and that's what we are doing now with very appealing results. We are also trying to find industrial partners now that would help us further develop the molecules and reach the first in-human tests. But that's another story, and that I will leave that for the next presentation, whenever that is. UPAB will certainly continue its efforts towards outreaching these activities. Before just thanking the people that actually did most of the work and the funders, I hope I have transmitted the idea that when we think about pandemics, we think about, doctors, nurses, healthcare professionals. We do not see at first glance biophysicists there being healthcare professionals, but they are certainly part of the process of protecting the lives of people in this situation. So this is mainly my main message. And we are working towards the next big pandemic. So I thank all the people in my group that worked in this research line. I also thank the EIC, the European Innovation Council, that funded most of the work, and our national funding agency, FCT. And thank you very much for your attention. I hope I was in time.
Thank you very much, Miguel. This was a really interesting talk. So this is the end of the session, Biophysics for Global Health and Sustainability. Thank you again to all our speakers. Thank you all for following the session. And now Arnab can take over. Thank you.
Thank you.
Thank you very
Thank
much.
you. Thank you, Christina. That was perfect. That was a series of fantastic talks. I wish you had kept some time for some question and answers. I'm sure there would be lots of very interesting questions, perhaps. Let me just add spotlight, and let me also add Narendra to the spotlight. Where is he?
Yeah.
Wait. There you are.
Yeah.
Okay, so great. It's nice to be back. Thank you, the biophysics panel. It was very, we learned a lot. I'm now going to hand over to my co-host, Narendra Deshmukh. He is a senior scientific officer at the Homi Bhabha Center for Science Education. Started off doing a lot of life sciences and then does a lot of educational research these days. And Narendra would be curating the next 2 talks, one from Mauritius, one from Sri Lanka, and also an activity that was done with students in a small town in Maharashtra, India. So over to you, Narendra. You can introduce yourself and then go ahead and start with Shakil's talk.
Thank you, Professor Arnab, coordinator slot 2, IYBSSD 2023, and Center Director, Homi Bhabha Center for Science Education. It's my pleasure and honor to introduce Dr. Shakil. Dr. Shakil has served the Mauritian education system for the past 23 years and is currently an academic in the Science Education Department of the Mauritius Institute of Education. Backed with a strong scientific and education academic background, he has shown his interest in science education, STEM education, quality assurance, and comparative studies. Through his publication, conference intervention, and research work, Dr. Shakil is currently the overall coordinator of a collaborative MIE and University of KwaZulu-Natal, Durban, South Africa project on STEM education. And he is also the international consultant for the quantitative analysis of the international project, that's COVID-19 impact on education system under the aegis of Stockholm University. Now, Dr. Shakil, would you like to say something before I ask Sonali, my colleague Sonali, to share the your clips? Dr. Shakil? Okay. Sonali, can
I think just go ahead and play the video.
Yeah, Sonali, can you play the video?
And you will have to speed it up by about 5% or so.
Yeah, sure.
In VLC, if you're doing that.
Greetings from the paradisiac island of Mauritius, a small island of the Indian Ocean, located on the southeast coast of Africa. These pictures showcase the island of Mauritius. I am Acha Shakil, from the Science Education Department of the Mauritius Institute of Education, which is a tertiary institution mandated for teacher education, curriculum development, and educational research. On the occasion of the International Year of Basic Sciences for Sustainable Development, I am pleased to unveil the theme of my intervention, which reads as follows: Basic Sciences as the driver of STEM education for sustainability. To understand the validity of this statement that basic sciences is the driver of STEM education for sustainability, I will drive you through 4 sections of this presentation. With the first one, STEM and STEM education, where we'll try to expose what is STEM and STEM education, together with its origin, evolution, and conceptualization. We'll then go on to the second part, STEM education for sustainability, to eventually focus on the real theme of my intervention, which is the role of basic sciences in STEM education for sustainability. We will eventually conclude and give time for discussion or question and answers. With that, let's start. What is STEM? This slide does not only show that the S of STEM stands for science, the T for technology, the E for engineering, and the M for mathematics. By the dots that you have in between those different disciplines, this picture depicts the origin and initial conception of STEM, which, backed by a political agenda at that time, had a career orientation, where the focus was on discrete disciplines, where the focus was on production of the workforce, that is to produce more scientists, more doctors, more ICT specialists, more engineers, among others. Let's now analyze the origin and evolution of STEM education after having described or defined the word STEM. Actually, from a political agenda, STEM finds its origin at the beginning of the 21st century in the United States, where the term SMET, S-M-E-T, was introduced by the National Science Foundation to emphasize on what we have been describing, that is on careers in science, technology, engineering, and mathematics, which were the need at that time. And in 2001, the American biologist Dr. Ramale rearranged the word to form the STEM acronym. So from SMET, S-M-E-T, it was now STEM, STEM. And from this point in time, the conceptualization of STEM, as shown in this slide, has moved from an enactment orientation to that of an interpretist orientation, to eventually give rise to the educational orientation, namely STEM education. Since then, STEM-focused curricula has extended beyond the frontiers of the United States to many other parts of the world, and is even now gaining a lot of traction, as STEM education is not only an approach, because STEM education prepares the youth for the future workforce. Actually, analysis of the literature revealed that the conceptualization of STEM education has been very dynamic over the past decades, accommodating different paradigm shifts. The first one was the shift from a STEM education where the focus was on teaching and learning of science, technology, engineering, and mathematics discretely as discrete disciplines to produce more scientists, IT specialists, engineers, and mathematics. Then it has moved to a different paradigm showcased by the two middle pictures of this slide, where there is a move in terms of how we bring those different disciplines together. That is, how we bring science, STEM, and science, technology, engineering, and mathematics together to form an interdisciplinary and integrated approach to teaching and learning. From this, indeed, we have been able to find a new conceptualization of STEM education. A conceptualization of STEM education where the traditional barriers erected between the four disciplines have been coalesced to form a cohesive, integrated, and holistic STEM education, where the disciplines are merged. This STEM education, this concept of STEM education, has not only brought the different disciplines together, but it has also opened doors for other disciplines. And today, we have different terms that are being used. When we are referring to STEM, it is not only about STEM. It may be a STEM with an additional M to integrate medicine in the equation of science, technology, engineering, and mathematics. Then we have STEAM with an additional A to integrate arts in the equation. STEAMed to integrate arts, education, and design. And most recently termed STREAM, where the R has two dimensions. In the first dimension, the R emphasizes on the integration of reading and writing in the equation of STEM, where we have the four disciplines: science, technology, engineering, and mathematics. In the second dimension, the R emphasizes on the integration of robotics, artificial intelligence, virtual reality, and augmented reality in STEM education. Having gone through those few slides, I believe that at least we have a basic knowledge of what is STEM, what is STEM education, what is the origin, the evolution, and different conceptualization of STEM education. Let's now focus on the STEM education for sustainability. Surely there is a difference between STEM education and STEM education for sustainability. In STEM education for sustainability, we are referring to an approach to teaching and learning that integrates the four different disciplines and any other additional discipline to develop not only knowledge, but skills, attitudes, and dispositions related to sustainable development or sustainability. As you know, sustainability in itself is an integrated approach where different perspectives, different factors are brought together towards sustainable development. And we need to have an education, an approach to teaching and learning, which is aligned to this concept of sustainability. Till now, one of the best approach towards teaching and learning of sustainability is the STEM education for sustainability. This approach aims to equip students with the necessary competencies needed to address real-world environmental challenges, climate change, etc., and to create a more sustainable societies. You will agree with me that many societies in the world today, we are facing a lot of issues, lot of challenges, and we really need an education which is aligned to what we are living in the society. which is relevant to what we are facing as challenges each and every day. So STEM education for sustainability involves teaching students about natural system, the impact of human activity on the environment, but it is not only limited to knowledge. It is about education that inspire, that empower learners to become responsible stewards of the planet and to make informed choices that will help them to create a more sustainable future for them and for the future generations. After the first two sections of my intervention, that is STEM and STEM education, followed by STEM education for sustainability, let's now focus on the theme of today. That is the role of basic sciences in STEM education for sustainability. In fact, in today's context, just like we have been discussing till now, STEM or STEAM or STEAMed or even STREAM, which are the integration of different disciplines such as science, technology, engineering, and mathematics together, are essential for the growth and development of any society. If we are referring to sustainability, we are directly referring to society and the benefits of having a society which has adopted innovation, progression, and development which is sustainable. In fact, STEM drive innovation, create new products and services, design and develop new processes and procedures, and address complex problems, which are key to the creation of a society based on sustainability. Reaching there, a reflection of this expected role of STEM education for sustainability brings me to a key question. What is the role of the basic sciences in the STEM education for sustainability? You will agree with me that for the benefit of the society and to move toward the future by adopting sustainability concept, there is no way out. We need to be able to adopt STEM education for sustainability. But it is much more important now to dissect this STEM education for sustainability, to understand what will be the role or what is the role of basic sciences as the driver of STEM education for sustainability. For the sake of this presentation, I will focus in my intervention on those 5 axes which are the role of basic sciences in STEM education for sustainability. The basic sciences is the driver because basic sciences provide foundation knowledge and understanding for STEM education to become a reality. The basic sciences is important because it's going to develop scientific literacy, which is key for STEM education and development of STEM skills. When we are referring to scientific literacy, when we are referring to development of competencies, skills, attitudes, all these go back to what we learn in science, in basic sciences, about inquiry learning, about the skills, the 21st century skills, the inquiry skill, the science process skills. All these skills are aligned to what we want in STEM education for sustainability. The basic sciences is also very important because it provides career opportunities and it is the basis of innovation and advancement. Therefore, the role of basic sciences in STEM education for sustainability is genuinely the driver of STEM education for sustainability. Let's take one by one now. As far as foundational knowledge and understanding is concerned, the basic sciences, which includes physics, chemistry, and biology, is not only important, but primordial for STEM education for sustainability. The basic sciences provide the foundational knowledge and understanding of how the natural world works, the interaction between biotic and abiotic factors within an ecosystem, the importance of addressing environmental challenges for sustainable future, and more importantly, the different concepts of sustainability. In fact, STEM fields, which include science, technology, engineering, and mathematics, are actually based on principles and concepts from physics, chemistry, biology, and mathematics. Without this foundational knowledge and understanding, no STEM initiative is possible. As a simple example, a lack of understanding of energy conversion as prior knowledge will surely prevent students from developing a model of photovoltaic system to produce electricity, which can be part of a STEM lesson or STEM project given at school. But without the prior knowledge on energy conversion, on types of energy, we cannot move to those aspects where we are bringing the different disciplines together. We need science. We need these basic sciences to understand, to be able to develop whatever complex concept in the teaching and learning. Secondly, the basic sciences also promote scientific literacy, which involves understanding scientific concepts and theories and being able to apply them to real-world problems. Students who study physics, chemistry, and biology gain an understanding of how the natural world works and how scientific research is conducted. And scientific literacy is essential for informed decision-making in today's world, where scientific issues like climate change, genetically modified organisms, and vaccines are hotly debated. This scientific literacy is very important for any layperson, for any person of the public, for any citizen of the society to be able to understand what is happening around you, to be able to understand the challenges we are facing in terms of emergence and re-emergence of epidemics, in terms of climate change. And all this can only be done by basic sciences providing scientific literacy. Then only we can bring this aspect into STEM lesson, STEM project, to showcase the importance of any concept. Thirdly, one of the main pillars of STEM education remains the development of STEM competencies among learners in view of transforming the lay public into informed citizens with the appropriate knowledge, skills, attitudes, values, and dispositions needed to address current and future challenges such as energy crisis, food crisis, climate change, emergence and re-emergence of epidemics, among others. These competencies, often referred as STEM skills, are actually the scientific process skills or inquiry skills. A strong foundation in basic sciences helps students to develop critical thinking and problem-solving abilities, among others. In fact, without a solid understanding of basic sciences and development of scientific skills, it would be difficult to excel in STEM fields and to make meaningful contributions to the advancement of a society. Let's now briefly explore the role of sciences in developing critical thinking and problem solving before moving to career opportunities and innovation and advancement. Focusing on critical thinking, a strong foundation in basic sciences is essential for developing critical thinking skills. Critical thinking involves analyzing information, evaluating evidence, making logical conclusions, among others, which are key scientific inquiry skills. In STEM fields, critical thinking skills are necessary for designing experiments, analyzing data, and solving complex problems. Moreover, students who have a solid understanding of basic sciences can apply critical thinking skills easily to any field they pursue. So this is what basic sciences is doing in terms of critical thinking, and thus in terms of development of competencies needed for the world of work for the future. Focusing on problem-solving abilities, basic sciences also provide students with different scenarios where they have to solve problems. A student who study physics, chemistry, and biology develop the ability to identify and analyze problems, generate hypotheses, and test them through experiments, generate and analyze data, infer and draw conclusions, and eventually recommend data-driven solutions to problems. These problem-solving skills are essential for any STEM field, where students have to find solutions to complex problems. Here again, the basic sciences is providing the ability to become problem solvers, which is key for the STEM field. Let's now focus on career opportunities. A solid foundation in basic sciences really opens up a wide range of career opportunities for students. STEM fields are projected to grow rapidly in the coming years, and there will be high demand for individuals with the skills in these areas. Students who study physics, chemistry, and biology and apply this in STEM lessons and projects can pursue careers in any field, be it medicine, engineering, big data analysis, artificial intelligence, computer science, scientific research, or any other STEM fields. In fact, the basic sciences provide the foundation for many high-paying careers in different fields, especially in STEM fields. The last part, innovation and advancement. Basic sciences are also essential for innovation and advancement, which is key component of STEM education. Scientific research in the fields like physics, chemistry, and biology has led to new technologies and products that have revolutionized the world. Without a solid foundation in basic sciences, it would be difficult to make meaningful contribution to scientific and technological advancement. We can always see the connection between the basic sciences and great advancement and innovation in the world. If I take the example of photovoltaic cell, what is it doing? It is capturing light energy and converting it into another form of energy. This is what plants are doing with the chlorophyll, with the different pigments, with the different photosynthetic pigment. They are capturing light energy and convert it into another form of energy. There are so many similarities, and it is those basic sciences which are the drivers of all these different innovation and advancement in our world. Let's take some examples of how science drives STEM. I will take one example in the field of medicine, one in the field of transportation, and one in the field of energy, though there are many more examples. The first example in the field of medicine. Medical science has made tremendous strides in the past century, leading to the development of vaccines, antibiotics, life-saving medical procedures that have improved public health and extended human lifespan. Medical research relies on scientific methods to understand the causes of diseases, develop new treatments, and test their efficacy through clinical trials. The application of scientific principles to medicine has led to a reduction in the morbidity and mortality rates and has improved the quality of life for millions of people worldwide. This would not have been possible without the foundational knowledge, understanding, and skills associated with basic sciences and which has been brought together in STEM education. Another example, in the field of transportation, the development of motor vehicles, airplanes, trains has revolutionized the way we move, we travel, and we transport goods. Transportation technology relies on scientific principles of motion, energy, and mechanics to design, build efficient and safe vehicles. The application of scientific principles to transportation in a STEM approach, bringing different other disciplines together, like mathematics, engineering, and also technology, has led to the growth of the automobile industry, the airline industry, and the logistic industry, which are critical components of global economy. Third one, in the field of energy, science has provided the foundation for the development of sustainable and renewable energy sources. The study of thermodynamics, fluid mechanics, and material science has led to the development of solar panels, wind turbines, and hydroelectric power plants. The application of scientific principles to energy has led to the reduction of greenhouse gas emissions, the promotion of energy independence, and the creation of new jobs in the renewable energy sector. You will agree with me that you name any discipline, we will be able to tell you that there are basic sciences and basic sciences principles and concepts which is driving the development, the innovation, whatever we have today, and we will be having tomorrow. So basic sciences is undoubtedly the driver of STEM, because STEM is the future of our society. STEM is about bringing everything together under an umbrella. And under this umbrella, the most important part which is driving, which is bringing the other discipline together, is science and will be science. On a concluding note, represented by this diagram, you can see that to drive this bicycle, we need basic sciences. So basic sciences is the driver of STEM education for sustainability. To conclude, the position of S in STEM is not a coincidence, as science drives the STEM approach, it drives STEM education, it drives innovation. Thank you very much. I remain available for any discussion, any comment. Thank you.
Thank you, Dr. Shakil, for a wonderful explanation about the role of basic sciences in STEM education. Thank you. Now, next session will be workshop, the workshop which we conducted at one of the school and one of the education college in Satara. Let me introduce my co-presenter, Dr. Sudhir Kumbhar. Dr. Sudhir Kumbar, working as a teacher at Red Education Society, Satara, Maharashtra. He is also a coordinator of Red Science Parishad. He is an environmental researcher. He also run environmental activities with students, youth, public since 1990. He's a writer of various articles on science, environment, and education. He is also working against a blind belief and matted hair removal. He more than 1,000 posters on nature he designed. Professor Dr. Sudhir Kumbar conducted the workshop with me at Red Education Society School and Education College. And also we have a workshop at Homi Baba Center for Science Education with practicing teachers. We have a recorded version of these videos. Basically, in this workshop, our focus was on plastic because as we are aware that today we are celebrating World Environmental Day, and the theme of our World Environmental Day is also plastic. So, considering the role of basic sciences in the context of sustainable development, we have chosen this topic for students, for school students, and also for student teacher and practicing teachers. I will also have to introduce Principal Vandana Nalaude ma'am and my colleague Sonali, because preparing workshop, designing materials and designing and making the video, a lot of resources, human resources as well as other resources also needed. So let me request Sonali, can you play that video, please? I will also explain the detail about this workshop, but let us see this video clips of this workshop.
आमच्या रयत शिक्षण संस्थेच्या महाराजा सयाजीराव विद्यालय सातारा प्लॅस्टिकचे दुष्परिणाम आपल्याला सध्या जगामध्ये भेडसावत आहेत आणि हे दुष्परिणाम आपल्याला कमी करण्याच्या संदर्भामध्ये आपण वेगवेगळ्या प्रकारचे प्रकल्प, उपक्रम, प्रयोग या विद्यालयामध्ये विद्यार्थ्यांना दाखवून दिले की जेणेकरून या विद्यार्थ्यांच्या माध्यमातून आज महाराष्ट्रामध्ये किंवा सातारा जिल्ह्यामध्ये याचं उद्बोधन होणार आहे, प्रबोधन केलं जाणार आहे प्रत्येक शाळांच्या मध्ये आमच्या रयतच्या, आणि त्याचा फायदा हा आपणाला पर्यावरण संवर्धनाच्यासाठी खूप चांगल्या पद्धतीचा होणार आहे.
तुम्हाला आम्ही हे जे काही इथे गट कार्य करण्यासाठी बोलवलेलं आहे या कार्यशाळेमध्ये, या मागचा हेतू असा आहे की आपण इंटरनॅशनल इयर ऑफ बेसिक सायन्सेस फॉर सस्टेनेबल डेव्हलपमेंट असं यावर्षीचं थीम आहे.
प्लास्टिक ही समस्या आहे. प्लास्टिकमुळे काय काय होतं कोण सांगू शकतो? प्लास्टिकमुळे काय समस्या आहे म्हणून? सिंगल यूज प्लास्टिक नावाचं प्लास्टिक आहे ना, समस्येमुळे आपला हवा प्रदूषण होते. आणखी प्लास्टिक अजून बोल. प्लास्टिक जनावरांच्या पोटात गेलं त्यांना. आता हे कशामुळे निर्माण झालं? प्लास्टिकची समस्या कशामुळे निर्माण झाली? हा राक्षस का तयार झाला? आपल्या ह्याचं नावच गेलेलं आपण प्लास्टिकचा राक्षस बाटली बंद करणे. बाटलीमध्ये त्या राक्षसाला कोंडून ठेवायचं आहे. आणि प्रयोग इथं तुम्ही करून बघणार आहे. त्याच्यामध्ये दोन तुम्हाला हे दाखवलेलं आहे. पहा इथं दोन, ह्याच्यामध्ये फरक काय दिसतोय का बघा? काय फरक आहे ह्याच्यात आणि ह्याच्यात? हे रंगीत प्लास्टिक आहे, बरोबर आहे, आणि ह्याच्यात ट्रान्सपरंट आहे. म्हणजे ज्यावेळी आपण सांगतोय कचरा वेगळा करायचा आहे त्यावेळी त्यांनी कसा गोळा केला पाहिजे ह्याच्या संदर्भात तुम्ही शेवटी सांगणार आहात. त्यानंतर इथं काही प्रयोग करायला हे ठेवलेलं आहे, म्हणजे मोजमाप घेऊ शकताय तुम्ही याचं. हे समजा असं आहे की असं विस्कटून बघितलं, आपण विस्कटून बघितलं टेबलावर, आपण विस्कटून टाकलं तर किती एरियात असेल काय ते गोळा केल्यानंतर कॉम्पॅक्ट केल्यानंतर ह्याच्यावर तुम्ही वजन करून बघा, तुम्ही किती प्लास्टिक गोळा केलेला आहे ह्या बॉटलमधलं वजन करून बघायचं. ठीक आहे.
बेसिक कॉन्सेप्ट शिकत असताना आपल्याला थोडासा विचार करायचा आहे, तो म्हणजे कुठेतरी सस्टेनेबल डेव्हलपमेंट. आणि यासोबतच आपण आपला जो काही दैनंदिन जीवनामध्ये वेगवेगळ्या गोष्टींचा वापर सुद्धा वाढत आहे. उदाहरणार्थ प्लास्टिक आपण चांगल्या मोठ्या प्रमाणात सगळीकडे वापरतो. म्हणजे आता तुम्ही इथे बघितलं तर इथे बऱ्यापैकी वस्तू काही प्लास्टिकच्या बनलेल्या आहेत, बरोबर. तर यावर्षी आंतरराष्ट्रीय पर्यावरण दिनाच्या निमित्ताने वर्ल्ड एन्व्हायरमेंटल डे जो आपण 5 जूनला सेलिब्रेट करतो, त्या दिवशी सुद्धा विषय जो घेतलेला आहे त्यांनी प्लास्टिक वरती विषय घेतलेला आहे. आपण जे काही शिकतोय, विज्ञानामध्ये शिकतोय, ते विज्ञान आणि ज्याला आपण शाश्वत विकास म्हणतो, सस्टेनेबल डेव्हलपमेंटला मराठीमध्ये शाश्वत विकास असं म्हटलं जातं. तर हा कसं आपल्याला साधल्या जाईल की पर्यावरणाचं समतोल पण राखलं गेलं पाहिजे आणि शाश्वत विकास सुद्धा झाला पाहिजे. तर त्या दृष्टीने आपल्याला हे समजून घ्यायचं आहे. आणि तुम्ही ऍज ए भावी नागरिक म्हणून, फ्युचर सिटीजन म्हणून तुम्हाला या गोष्टीचा जास्त विचार करावा लागणार आहे. तर त्या दृष्टीने आपल्याला एक जनजागृती कशी करता येईल समाजामध्ये किंवा आपल्या आजूबाजूला.
आणि तुम्ही विद्यार्थी इथे तुम्हाला दिसतंय 3 आर तत्वांचा वापर करणे. 3 आर तत्व आपल्याला माहिती आहे, रिड्यूस आहे, रियूज आहे, रिसायकल आहे. आणि त्या तत्वांचा वापर करून इथं जे आहे पहिल्यांदा तुम्हाला वर्गीकरण कचऱ्याचं करता येते का ते आम्ही बघतो. वर्गीकरण करत असताना प्रत्येकाला एक तक्ता आपण देतो आणि त्याप्रमाणे तिथलं साहित्य काढायचं तिथं ठेवायचं. आणि 3 आरचा तुम्ही हे कसं करता, 3 आर कसं वापरणार, रिड्यूस कसं करणार या बाबतीत, रिसायकल कसं करणार या बाबतीत, किंवा रियूज कसा करणार त्याच्यावर चर्चा करणार आहेत. आणि 1-1 मिनिटांमध्ये तुम्ही सांगायचं त्यातलं महत्वाचं काय असेल. काय पाहिजे, रिड्यूस म्हणजे कमी करा, रियूज म्हणजे पुन्हा पुन्हा वापरा, आणि रिसायकल म्हणजे पुनर्चक्रीकरणासाठी कसं देता येऊ शकेल. पहिली गोष्ट आहे ई वेस्ट. ई वेस्ट मी इथं देतो, ई वेस्ट. दुसरा आहे प्लास्टिक, प्लास्टिक वाल्यांना इथं देतो. ग्लास, काच आहे. त्यानंतर थर्माकोल, त्यानंतर मेटल आणि पेपर. इथं यायचं आहे आणि इथलं साहित्य त्यात एक दोघांनी घेऊन जायचं, कळतंय. आणि तिथं गेल्यानंतर तुम्ही चर्चा काय करणार आहात, 3 आर तत्वाचा वापर मला करायचा न पडता कासा वगैरे आहे तर आहे काळजी घेते. आता तुम्ही सांगा बंद करताना तुम्हाला काय जाणवलं किंवा काय वाटते तुम्हाला ही ऍक्टिव्हिटी केल्यानंतर.
जास्त प्रमाणात जागा लागतं, पण जर आपण ते गोळा करून एका बाटलीत बंद केलं तर ते कमी जागा लागतं आणि प्रदूषणच नाही.
समजा हे असं ते एकदम पॅक केलेलं असं दुसरी लावली, तिसरी गेलं किंवा ह्याच्यामध्ये हे वापरणार का हे वापरणार, कोणतं का वापरणार, मजबूत आहे. म्हणजे तुम्हाला ज्यावेळी भरायचं आहे त्यावेळी कसं भरणार आहे, तुम्ही ठसून भरायला, बरोबर आहे. आणि मग तुम्हाला त्याचा बीज म्हणून वापर करता येऊ शकेल. बरं, नाही संकलित केलं त्याचं काय करायचं असं माझं म्हणणं आहे. बरं, शो साठी वापरतो. बरं, अजून सिंगल यूज प्लास्टिक आहे. सिंगल यूज प्लास्टिक धोकादायक आहे. खुर्ची तयार केली किंवा प्लास्टिकचं काय तयार केलं दीर्घकाळ वापरणार, पण सिंगल एकदा वापरायचं आणि फेकून द्यायचं. कुरकुरेचा कागद असेल किंवा अन्य काय असेल. इथं एक काय केले बघा, असं काय करता येते का? असं करता येईल. दुसरी गोष्ट, मला 3 R म्हणजे रियूज, रिड्यूस, रिसायकल करायचं. तर रिसायकलला द्यायचं असेल तर कुणाला द्यायचं? कोण येणार? तुमचं काय नाव? मोठं प्लास्टिक मगाशी बघितलं, एवढं सगळं अंतरलं होतं. किती बाय किती होतं ते? 370. आणि आता किती झालं? 51 ग्रॅम. लांबी रुंदीला किती? आता लांबी रुंदी अंदाजे सांगा.
एक अंदाजे सांगा.
10 गुणिले 5 50. आणि उंची किती आहे? उंची 250 घन सेंटीमीटर मध्ये मावला तो पसरा. किती पसरला होता? तुमच्या प्रत्येकाच्या घरामध्ये असं काही करायचं ठरवलं तर तुम्ही कसं करा? तुम्ही काय करा? आता तुम्हाला सुचलेली आयडिया सांगायची. एकेका गटाने एक आयडिया सांगा. काय सुचेल तुम्हाला? असं मी करू शकतो, असं मी गावात करू शकतो.
सुद्धा नवीन आयडिया देऊ शकता.
तुम्ही उजळवणार कसं? तुमच्याकडे यंत्रणा आहे का तेवढी?
कुंडाचा आकारमध्ये ते ठेवून त्याच्यात म्हणजे कुंडाच्या साच्यामध्ये त्या बॉटल ठेवून त्याच्यामध्ये सिमेंटचं भरायचं कलरने आणि कुंड्या तयार करायच्या.
छान. कुंड्या तयार करता येईल, कट्टा तयार करता येईल किंवा आणखी काही गोष्टी तयार करता. अजून काय? डोळ्यापुढे फक्त सिंगल यूज प्लास्टिक ठेवा. बंधन आणायचं आहे. कसं बंधन आणाल तुम्ही? प्लास्टिकला बंधन आणायचं त्या किंवा लोक वापरणार आहेत, पण त्याच्यावर तुमची उपाययोजना काय? तुम्ही अधिकारी झाला तर तुम्ही काय करणार? पोलीस झाला तर काय करणार? न्याय व्यवस्थेमध्ये गेला तर काय करणार? वन खात्यात झाला तर काय करणार? डॉक्टर असला तर काय करणार? शिक्षक असला, सामान्य नागरिक, शेतकरी असला म्हणून काय करणार? असल्या प्लास्टिक एका ह्याच्यामध्ये सांगायचं.
पहिल्यांदा स्वतः वापर टाळणार आणि मग दुसऱ्यांना टाळायला सांगणार.
प्लास्टिकच्या वापरासाठी म्हणजे जसं आपण प्लास्टिकच्या पिशव्या वापरतो त्याऐवजी कापडी पिशव्यांचा वापर करू. आणि ज्या ठिकाणी लिक्विड स्वरूपात आपण वस्तू वापरतो त्यामध्ये प्लास्टिकच्या ऐवजी स्टील किंवा इतर धातू वापरण्यासाठी वापरत आणू. आणि ज्या ठिकाणी त्याच्याऐवजी वेगवेगळ्या वस्तू बनवतात तिथे सुद्धा धातूचा वापर वाढतो.
रिसायकल सरांनी बरंच सांगितलं, पण रिसायकल करत असताना सुद्धा आपल्याला एक विचार करावा लागणार आहे की रिसायकलिंग प्रोसेस सुद्धा प्रदूषण करणारी नसावी. तो सुद्धा एक आपल्याला शास्त्रीय सायंटिफिकली विचार केला पाहिजे की रिसायकलिंग कशा पद्धतीने असलं पाहिजे लक्षात आलं. आणि रियूज कडे कसा भर देता येईल.
थर्मोकोल कसं तयार करतात किंवा ते कसं तयार कसं करतात तुझंच नाही फेकून देतो आपण. आणि आपल्याला असेल किंवा कप असतील फेकून देतो. तुमचं म्हणणं सांगा.
थर्मोकोलचा वापर बघितला तर दुकानामध्ये होतो, घरात डेकोरेशन करायचं म्हणतो, परत फंक्शन्समध्ये होतो. आणि ट्रान्सपोर्टेशन त्याच्यात मोठ्या प्रमाणात कारण की अँटिक पीस किंवा नाजूक वस्तू असतात त्यांना डॅमेज होणार त्यासाठी याचा वापर होतो. परत दुकानात देखील त्यासाठीच होतो आणि डेकोरेशनमध्ये ह्याचा वापर होतो. तर हे सहसा तर डिकम्पोज होत नाही कारण आणि याचा मेल्टिंग पॉईंट सगळ्यात कमी होते. त्यामुळे जळतं आणि जळल्यामुळे कार्बन डायऑक्साईडचा
उपयोग काय करता येईल नंतर?
याचा उपयोग म्हणलं तर काय करतात डेकोरेशनसाठी
अजून एक मध्ये काहीतरी काढलं होतं बघा, इथर मध्ये किंवा बेन्झिन मध्ये हे टाकायचं आणि त्याचं सोल्युशन तयार करताना की असं दिलं तर ते वॉटरप्रूफ तयार असतं.
लाकडावर पॉलिश
हा
पण हे डांबरमध्ये मिक्स केलं
होऊ शकतं. प्रयोग करून बघायला पाहिजे तुम्हाला.
ते पडलं नाही म्हणजे डॅमेज तर पडणार नाही.
यात जर विरघळलं तर खड्डा पडणार. जर तसं केलं तर विरघळलं तर जे थर्मोकोल जे आहे थर्मोकोलचं करता येणार नाही, त्याचं करता येऊ शकेल ना, ते पॉलिइथिलीन फोमच आहे. टाकूनच दिलं जातं, कृत्रिमरित्या तयार केलं केमिकल, हे केमिकल आहे, तर ते केमिकल वातावरणामध्ये गेलं तर धोकादायक आहे. त्याचा वापर करायचा का नाही तुम्ही ठरवायचा आहे. थर्मोकोलचा कप घ्यायचा का नाही.
प्लास्टिकच्या माणसाने शोध लावले, त्याला वाटतं की आपल्याला खूप चांगलं ठरेल, पण मग त्यांना ऊस गोड लागला म्हणून वास होतो. जसा जसा माणसाला प्लास्टिकचा वापर वाढवायला सुरुवात केली तसं तसं ते तिला फार घातक ठरत आलं. आपण रोज प्लास्टिकच्या पाण्याच्या बाटल्या वापरतो, आपण हे पाणी पितो आणि कुठे रस्त्यावर फेकून देतो, पण हे आपण प्रॉपर ह्याला रिसायकल करत नाही. तर ह्या रिसायकल मेथडमध्ये इथून येतात. आपण हे रोज पेन वापरतो, पेन किती 5 रुपयाला असते, हे शिल्लक समजून आपण हे असे संपले की आपण फेकून देतो, पण ह्याच्यामध्ये आपण 2 रुपयाची रिफिल टाकू, ह्याला पुन्हा वापर करू शकतो. आणि ह्या प्लॅस्टिकच्या बॅग आपण कुठं भाजी घ्यायला गेलो, कुठं गेलो तर आपण सगळ्यात पहिलं होतो प्लॅस्टिक बॅग, पण आपण असं नाही केलं पाहिजे. तर आपण स्वतःची आपली कापडी पिशवी, कागदी पिशवी ह्याचा जास्तीत जास्त वापर केला पाहिजे आणि ह्याला रिड्यूस केलं पाहिजे हळूहळू. ही बॉटल, ह्या बॉटल आपण संपल्या की असं कचऱ्याच्या डब्यात टाकतो, कारण ह्या आपल्याला शुल्क वाटतात, पण ह्या पुढे जाऊन आपल्या पर्यावरणाला फार घातक ठरतात.
कमीत कमी कसं होईल, त्याचा पुनर्वापर होईल का आणि ते पुन्हा पुन्हा वापरता येऊ शकेल का, पुनर्चक्रीकरणासाठी देता येईल का, हा उद्देश आजच्या ऍक्टिव्हिटीचा. या ऍक्टिव्हिटीमधनं तुम्हाला जो बोध झाला तो समाजापर्यंत पाठवण्याचं काम तुम्ही कराल अशी अपेक्षा व्यक्त करतो. तुम्ही वेगवेगळ्या शाळेतून आलेला आहात, तर त्या शाळांच्या मध्ये असा प्रकल्प राबवा की वर्षभरामध्ये 400-500 बॉटल्स, एका विद्यार्थ्याने एक बॉटल तयार केली चालेल, तो सगळा कचरा जो पसरणार होता, अख्ख्या गावभर पसरणार होता, शेतामधनं किंवा नदीमधनं ओढ्यामधनं तो समुद्रापर्यंत जाणार होता, तो तुम्ही थांबवण्याचं काम कराल अशी अपेक्षा व्यक्त करतो आणि थांबतो. पुन्हा एकदा तुम्हाला पर्यावरण दिनाच्या हार्दिक हार्दिक शुभेच्छा.
Now, second workshop we conducted for B.Ed student teachers.
रयत शिक्षण संस्थेच्या आझाद कॉलेज ऑफ एज्युकेशन सातारा समाजाचा आपण एक भाग आहोत आणि समाजासाठी आपण काय काय करू शकतो, पर्यावरणासाठी काय काय करू शकतो, यासाठीही काही त्यांना कृती दिल्या जातात. आज UNEPच्या प्रोग्रामच्या निमित्तानं आमच्या विद्यार्थ्यांना आम्ही प्लास्टिक संकलन करून आजूबाजूच्या परिसरामध्ये लोकांच्या मध्ये प्लास्टिक निर्मूलनासंदर्भातली जनजागृती करण्यासाठी आम्ही त्यांना सांगितलं होतं. त्याचबरोबर ते कलेक्ट करून प्लास्टिकच्या बॉटलमध्ये जास्तीत जास्त प्लास्टिक भरून आणून महाविद्यालयामध्ये त्याचा पुनर्वापर करण्याच्या दृष्टिकोनातून आपण प्रयत्न करत होतो.
काही ऍक्टिव्हिटी घेतल्या विद्यार्थ्यांच्या दृष्टिकोनातून पर्यावरण कसं पाहिजे. तुम्हाला गट आम्ही देतोय, एक आहे ई वेस्ट, दुसरा आहे पेपर्स आणि रद्दी, तिसरा आहे धातू दिले जातील, चौथा आहे थर्मोकॉल, पाचवा आहे काच आणि 6वा आहे ई वेस्ट.
आपण त्याच्यातलं बघायचं पेपर मध्ये कोणता आहे, या गटाने त्याच्यामध्ये धातू कोणता आहे,
पेपर आणि रद्दी, धातू धातू हे घेऊन जा, थर्मोकोल आणि अनुषंगिक दोन्ही पण चर्चा करायची पण त्याच्यावर. पॉलिथिलीन फोम नावाचं फोम असतं. ई कचरा पुन्हा काय, ई कचरा एक
अच्छा वर्गीकरण तुम्ही केलेलं आहे का, तुम्ही हे केलेलं आहे
फक्त त्यांनी सांगायचं खाली. तर जास्त जागा व्यापली त्याच्याऐवजी तो कॉम्प्रेस करायचा आहे आणि याच्यावर वजन करून बघायचं आहे किती जागेमध्ये मावतो. डायमेन्शन, लांबी, रुंदी, उंची आता आपण मांडून ठेवा. एकदा वापरण्यात येणारा आणि थ्रो बिस्किटचा पुढचा कागद वारंवार वापर. खुर्ची घेतली तर खुर्ची मी 2-4 वर्ष 10 वर्ष पण वापरू शकतो. बघ नुसतं लागू मुद्दाम करायला इकडे बघा किती ग्रॅम ग हे
285 आणि 226.
आता हे जे आहे बांधकामामध्ये कसं वापरता येईल, सिमेंट आणि एका बाजूला तुम्हाला डिझाईन असलं पाहिजे असेल तर असे सगळ्या दिसते, पण आपल्याकडे पोरं आहे की बुजं काढणार, मग काय करायचं त्या बाजूला आणि त्याच्यामध्ये परत माती भरल्यानंतर तुम्हाला त्याच्यामध्ये सांग मोड करता येते आणि पाहिजे तेवढं कट्टा तयार करता येतो. कट्टा तयार झाला की त्याला प्लास्टर करता येते.
तर पॉलिस्टिरीन या पदार्थापासून थर्मोकोल तयार केलं जातं. थर्मोकोलचा वापर कुठे कुठे केला जातो तर काचेच्या वस्तू कॅरी करण्यासाठी किंवा फ्रीज, टीव्ही या वस्तू कॅरी करण्यासाठी, त्या वस्तू खराब होऊ नयेत किंवा फुटू नयेत म्हणून थर्मोकोलचा वापर केला जातो. थर्मोकोल हे निसर्गासाठी हानिकारक आहे, त्याच्यामुळे त्याचा रिसायकल होतं पण रियूज त्याचा करू नये असं आहे. रियूज करताना मग थोड्याशा नोबेच्या वस्तू त्याच्या बनवल्या असतात, त्यापासून मी शो पीस असं झुंबर बनवलाय, त्याच्यामध्ये लाइटिंग सोडली, मस्त दिसते.
प्लास्टिकचा वापर रियूज केला. 3 R आहेत बघा, रिड्यूस, रियूज आणि रिसायकल.
मोबाईल, लॅपटॉप, पेनड्राईव्ह हे आपण जे वापरतो आणि त्याचा जो कचरा होतो त्याला आपण ई वेस्ट म्हणतो. तर आपण तेही कसं करायचं बघा, रिड्यूस. जेव्हा आपला मोबाईल खराब होतो, आपला मोबाईल खराब झाला तर आपण दुरुस्त करून तो वापरू शकतो. किंवा आपण जर मोबाईल नवीन घेतला तर आपला जुना मोबाईल आहे तो जुना मोबाईल आपण पुन्हा एका गरजू व्यक्तीला देऊ शकतो किंवा एखाद्या शाळेला लॅपटॉप भेट देऊ शकतो.
हा इसवी सन 300 ते 200 वर्षापूर्वी इजिप्तला झालेला आहे आणि सिलिकॉन आणि मायटर या दोन्ही संयुगाची क्रिया होऊन काच तयार झाली असं लिहिलेलं आहे, म्हणजे आपल्याला असं सापडतं. तर रिड्यूस आपल्याला काचेचा कसा करता येईल, काचेची भांडी आपण वापरतो, घरी बाउल्स वगैरे वापरतो, तर त्याऐवजी आपण मातीची भांडी वापरू शकतो. काचेचा रिड्यूस आपण असा करू शकतो, म्हणजे काचेच्या प्लेट्स असतील किंवा काचेचे बाउल्स असतील तर त्याच्याऐवजी आपण मातीच्या प्लेट्स आणि मातीचे बाउल्स आपण वापरू शकतो. रियूज काचेचा असं करता येईल की आपण ज्या बॉटल्स काचेच्या फेकून देतो त्या फेकण्याऐवजी त्याच्यामध्ये आपण प्लांट्स आहेत, शोचे प्लांट्स घरगुती आपण ठेवतो बघा, मनी प्लांट्स वगैरे, तर तशी सुद्धा आपण काचेच्या बॉटल्समध्ये आपण घरी
त्याच्यामध्ये तुम्ही त्या तुमच्या क्रिएटिव्ह आयडिया कागदावर टाकू शकता. आपण त्याच्यावरती चर्चा करू शकतो आणि तुम्हाला वेगळी दिशा सुद्धा देऊ शकतो. त्याला विज्ञानाचाच विद्यार्थी असं नाही, आपल्या आयुष्यात सगळ्यांच्या आयुष्यामध्ये विज्ञान आहे प्रत्येक गोष्टीत. त्यामुळे असं त्याच्यात कुठलंही बंधन नाही की लँग्वेजचा त्यांनी करायचं नाही, सायन्सचा त्यांनीच करायचं असं नाही. आपण सगळेजण सायंटिफिक ॲटिट्यूड असणारे शिक्षक असायला हवेत आणि त्यासाठी आमची ही धडपड आहे. आपण सगळेजण त्याच्यामध्ये सहभागी होऊया.
भूमिभाव सेंटर आणि रयत विज्ञान परिषदेच्या वतीनं या परिसरामध्ये 2 कार्यशाळा आज घेण्यात आल्या पर्यावरण दिनानिमित्त. त्या कार्यशाळेमध्ये विद्यार्थी आणि विद्यार्थी शिक्षक बीएड कॉलेजचे त्याच्यामध्ये सहभागी झाले होते. या सगळ्यामध्ये 3R म्हणजे रिड्यूस, रियूज आणि रिसायकल याचा वापर कसा विद्यार्थी करतात किंवा समाजासाठी त्याचा उपयोग कसा होऊ शकतो पर्यावरणाचं प्रदूषण रोखण्यासाठी, हे एक ॲक्टिव्हिटी होती. आणि दुसरी ॲक्टिव्हिटी होती की जो प्लास्टिकचा सिंगल यूज प्लास्टिक आहे त्याचं रूपांतर करणे आणि एकत्रित हे प्लास्टिक करता येईल आणि बाटली बंद करता येईल यासाठी ही ॲक्टिव्हिटी घेण्यात आली. याच्यामध्ये विद्यार्थ्यांनी अत्यंत चांगल्या पद्धतीनं सहभाग घेतला आणि प्रबोधनामध्ये त्याचा उपयोग करण्याचं त्यांनी ठरवलं.
आता हे भावी शिक्षक असल्यामुळे त्यांच्यामध्ये ह्या गोष्टी जेव्हा त्यांच्यामध्ये रुजल्या जातील तेव्हा आपोआपच त्यांच्या हाताखालून जाणारे हजारो विद्यार्थी पर्यावरणाचा योग्य दृष्टिकोन असणारे तयार होतील आणि त्यासाठी ही ॲक्टिव्हिटी, ह्या कृती खूप गरजेच्या होत्या.
नमस्कार. आपण आज रयत एज्युकेशन सोसायटीच्या शाळेमध्ये आणि आझाद शिक्षण महाविद्यालयामध्ये 2 कार्यशाळा आयोजित केल्या होत्या. या कार्यशाळा आयोजित करण्यामागचा हेतू असा होता की इंटरनॅशनल इयर ऑफ बेसिक सायन्सेस फॉर सस्टेनेबल डेव्हलपमेंट हा जो काही युनेस्कोचा प्रोग्राम आहे आणि दुसरं म्हणजे वर्ल्ड एन्व्हायरमेंटल डे त्या निमित्ताने 5 जूनच्या निमित्ताने हा IYBSSD 2022-23 हा प्रोग्राम सादर केला जाणार आहे. त्यामध्ये आपला प्रयत्न असा होता की शिक्षक, विद्यार्थी आणि शिक्षक विद्यार्थी त्यांचा जो काही शाश्वत विकासाच्या संदर्भात आणि मूलभूत विज्ञानाच्या संदर्भात जे काही ते सध्या कार्य करत आहेत ते जनसामान्यापर्यंत पोहोचावं हा या मागचा हेतू होता. आणि टाटा इन्स्टिट्यूट ऑफ फंडामेंटल रिसर्च ही संस्था या कार्यामध्ये सहभागी आहे आणि एक प्रतिनिधी म्हणून TIFRचा आणि रयत शिक्षण संस्थेचे डॉक्टर सुधीर कुंभार आम्ही दोघांनी मिळून ही कार्यशाळा 2 ठिकाणी
दिस
We also discussed with practicing teacher on plastic pollution. Thank you. We would like to thank Tanmay and Priyanka Patil, and also we would like to thank Vinita and my colleague Sonali for wonderful support. In this workshop, basically, as we have seen that we tried to address 2 objectives. The first objective was to promote an understanding of how to maintain a balance in the environment and achieve sustainable development. And second objective was to explore ways to create awareness in society regarding the pollution caused by plastic and to make them aware of the working of students and student teacher about sustainable development and basic sciences. Thank you. Now we have a session, another session by Professor Kokila Konasinghe. She is a professor in law and the head of the Department of Public and International Law at the Faculty of Law, University of Colombo, Sri Lanka. She is the coordinator of the MPhil and PhD programs and the founding director of Center for Environmental Law and Policy at the Faculty of Law, University of Colombo. She is a member of the Commonwealth Future Climate Research Cohort 2021 and 2022 of the Association of Commonwealth Universities and has represented Sri Lankan official delegation at the United Nations Climate Change COP27 in Egypt. Professor Konasinghe involved in 2 SAARC regional level projects representing Sri Lanka, the United Nations Millennium Fellowship Program for producing the South Asian report on climate change and preparing the country report on international disaster management, laws and policies under a collaborative project with other SAARC countries and International Red Cross and Red Crescent Society. Professor Kokila is the former director of the International Office of the University of Colombo and the president of the Art Council of the University of Colombo. She was a visiting researcher and a teaching fellow at Erasmus University, Netherlands, University of Granada, Spain, and Jindal Global University, India. She collaborates many research programs with the Special Center for Disaster Research at Jawaharlal Nehru University in India. She is a teaching fellow of Maharashtra National Law University, Mumbai, India. Professor Konsinghe is a chair in South Asian Postgraduate Law Conference, which covers all 8 countries of the region of South Asia. Her book chapter and research articles have widely appeared in locally and internationally reputed journals and publications. Now, let me play Dr. Kokila's views on law and environment.
Since
in the
Kokila is here, do
context.
you want to ask her to say a few words?
Yeah, I think
Play
so.
the video? I mean, she's here, so I know
yeah,
she's not well. Thank you so
that's-
much for joining us in spite of your ill health. We will play your video. We will not make you give a 20-minute, 25-minute talk. But if you just like to say something for 30 seconds, 1 minute, we would really appreciate it.
Okay, thank you so much, Aiboan. Greetings from Colombo, and it's namaskar. It's lovely to join with you all, even though I'm unwell. So I wish I could present live, but can't hold myself. So thank you so much for giving me opportunity, and thank you UNESCO and Tata Institute of Fundamental Research. Thank you.
Okay, let's have the video then.
Hi, Boan. Greetings from Colombo, Sri Lanka. I'm Professor Kokila Konasinghe, and I'm the head of the Department of Public and International Law Department. At the Faculty of Law, University of Colombo, Sri Lanka. And I teach environmental law and human rights law for
Sonali.
undergraduate and postgraduate students.
Yes, sir.
I was a member of the
Can you make it full screen?
Climate Research Cohort 2021 to 2022. And I was fortunate enough to represent Sri Lanka at COP27 last year. I'm the founding director of Center for Environmental Law and Policy, CELP, at the Faculty of Law, University of Colombo. I have engaged in research at the regional level on climate change and also on disaster management laws. And also, I was a visiting researcher and a teaching fellow at Erasmus University Rotterdam in the Netherlands, and also University of Granada in Spain. I was a research fellow in Jinda Global University, India, and also I collaborate many research programs with Special Center for Disaster Research at Jawaharlal Nehru University in India, and also Maharashtra National Law University and South Asian University in India. So thank you very much for joining with me today. And my speech on this World Environment Day presentation is on beyond reasonable doubt, the relevancy of basic sciences in improving the rights of nature. I consider this opportunity to talk to you on the World Environment Day as a divine task pressed upon me. Why? Not because I am an environmental law expert, but because I am fortunate enough to continue to survive on this planet today, despite the detrimental environmental catastrophes pose threat on the earth. We, the mankind, are the worst destructors of Mother Earth. Therefore, my main focus is to analyze why we should go beyond claiming human right to environment, to celebrate rights of nature, and in order to do so, what support the legal experts seek from basic sciences and its application. The right discourse between Homo sapiens and nature has been evolved since the ancient time till to date. Trace it back in the Old Testaments, Adam and Eve were tempted by the Satan to eat fruits from the forbidden tree, and for what they were cursed by the God. One can argue this story resembles that the human greed will destroy nature and subsequently destroy human survival itself. Overconsumption of natural resources by the present generation poses threats for the future generations of enjoying the same fruits of natural resources for their development needs. According to ancient scriptures of Mahavansa, in 600 BC in Sri Lanka, where Arhat Mahinda, who was in diplomatic religious mission to Sri Lanka from India, preached to the Sri Lankan king that all wild beasts have the right to live. The king is only a ruler and the custodian. Nature has its own rights and the ruler cannot determine its ownership. Determination of superiority of nature and human beings is an eternal struggle since the time immemorial. The legal approaches to protect the environment have so far been biased towards human-centered focus, i.e. anthropocentric approach, which emphasize the protection of rights of human beings towards the environment. On the other hand, much broader and a holistic approach to protect the environment is ecocentric focus, which is inclusive of humans and nature, the entire spectrum of the earth. The discussion on anthropocentrism and ecocentrism open another debate in the rights discourse as human right to environment and rights of nature. If the environment is not clean or healthy, then the humans will also face unsatisfactory living conditions. For instance, according to the Special Rapporteur on Human Rights and the Environment of the United Nations Office of the High Commissioner for Human Rights, a healthy environment is integral to the full enjoyment of wide range of human rights, including the rights to life, health, food, water, and sanitation. Accordingly, the right to environment relates to the entitlement of a person to live in better environment standards. However, contemporary environmental challenges which disturb a number of ecosystems, such as climate emergency, disappearing environmental zones, unselective felling of trees, expanding of deserts, various actions, damages to the environment may not be able to resolve, only be recognizing them by human rights violations. Therefore, all integral parts of the global biodiversity due to all ecosystems require to be separately protected as integral parts of the global biodiversity. Due to these concerns, the human-centric approach of protecting environment through the right to environment is now evolving into a broader notion of rights of nature, which is considered as the ecocentric approach of environmental conservation. The rights of nature reevaluates human rights and relationships with nature. In the most simple meaning, it strives for a transition in which nature is placed at the core and the humans are connected to it in an independent way, as opposed to the right to environment, which was dominatedly human-centric. Most importantly, rights of nature identifies that an ecosystem has the right to exist, flourish, regenerate its vital cycles, and naturally evolve without human-caused destruction. Accordingly, it provides subsidiary rights of nature, such as right to restoration, right to natural processes, right to ecosystem functioning without interference. The rights of nature grant ecosystems the right to legal personhood and the right to legal representation by a guardian. The right to legal personhood entitles ecosystems to defend themselves in court against any human-induced or nature-induced destruction or degradation. Rights of nature activists have utilized various avenues within the justice system and legal remedies to further develop this concept. These avenues include constitutional and statutory provisions, judicial review, and green activism, including indigenous movement. The doctrine of rights of nature is gaining momentum across the legal world. A series of cases were filed as legal experiments. Some were successful, while others failed. A careful analysis of the reasons for failure or success notes that the lack of scientific establishment of the legal personhood largely contributes to making fruitless claims under the rights of nature. Therefore, basic sciences and its data play a vital role in proving the logical sequences of facts when proving a case on rights of nature. The most related point of my discussion for today's presentation, that is how we can use basic sciences in establishing the argument on rights of nature more correctly based on scientific evidence, is as follows. The absence of evidence to prove the link between damage and the main causes is one of the key defensive arguments against the rights of nature. Another defense against rights of nature is the notion that the nature cannot embrace rights and responsibilities equally. In many jurisdictions where the rights of nature claims have succeeded, reliance on scientific data and findings has been substantial. Therefore, legal arguments seeking justice for nature need to be supported with well-established scientific data. In analyzing the relevancy of basic sciences in proving the legal arguments of rights of nature, we can identify several unanswered questions in the legal domain. Some of the key elements of those questions are as follows. Number one, how to define nature? The definition of nature can be varied depending on the subject in the matter of concern. The nature can be defined according to the sociological, political, geographical, spiritual, and aesthetic sense. Therefore, how nature can be defined according to the available scientific evidence and analysis could be the closest definition to universal acceptance. Basic sciences can address the issue of definition with the minimum confusion. Number two, how may nature claim its rights? The definition of nature plays a significant role in determining how it claims rights. So far, the legal assessment on how to claim rights can be either the government agents or custodian or a guardian can act on behalf in claiming the rights on behalf of nature. Number three, what rights nature will have? Nature can have moral rights and legal rights. Its legal rights derive from constitutional principles like in Ecuador Constitution. statutory provisions like in Whanganui River Settlement Act in New Zealand, or from judicial precedents like in India and Bangladesh. Rights of nature has been identified as natural communities and ecosystems who exist, flourish, and naturally evolve. However, determining the scope of its rights, the use of basic sciences can be very useful. Objective interpretation of the relevant parameters in determining what rights can nature claim would be a fascinating scientific finding for legal interpretation. Number 4, how can nature be held in responsibility? In Lalit Miglani and Mohammad Salim, the two famous landmark Uttarakhand High Court judgments in India, the defense lawyer raised the issue of held the nature responsible for damages caused in natural disasters. Under the legal and political principles such as representative democracy and state responsibility, states, the government, can be held in responsible for human rights violation. Example, state shall take measures to prevent systematic torture. Therefore, the government agents under whose purview the necessary preventive mechanisms and actions should be held responsible for the natural disasters on behalf of the nature. In order to take necessary preventive actions, basic sciences such as geology, physics, and other related subject areas can be beneficial in weather forecasting and geographical calculations and measurements. Number 5, the traditional locus standi is granted only to natural persons and legal persons. Rights of nature scholars advocate that the legal personhood can be changed to legal naturehood to avoid confusions related to using similar yardsticks. Those have been used in determining legal personhood in natural objects. In winding up the entire argument, I would say 3 components. First, the nature is facing a complex and challenging situation today with the unsustainable manner of human interference in natural evolution with the short-term development goals. Existing legal frameworks in regulating this human interaction has been limited to anthropocentric strategies. Therefore, the nature needs a broader and more inclusive strategies upon facing the complex challenges. Ecocentric approach in environmental law has been recognized as a strategy in addressing complexities in global environmental problems. Secondly, rights-based approach in environmental protection in connection with the third generational human rights discourse took considerable effort to prevent environmental damages that cause damages for a clean and healthy environment and also maintain a quality living standard in the environment that is sustainable for human survival. After a careful assessment of human right to clean environment, the scholars have now realized that human rights alone cannot solve the global environment problem that can adversely impact nature itself. Therefore, the novel approach is right-based path is rights of nature. Finally, in concluding, in order to establish more acceptable solutions for global environmental problems, the nature should be looked at from non-traditional legal parameters. Therefore, rights of nature scholars suggest legal naturehood in place of legal personhood for the justice seekers who come to courts. In order to place the argument of nature as a legal person before the court, basic sciences can play a main role. Many arguments in favor of establishing rights of natural phenomenon to be protected and flourished should be proven with scientific data and findings. Scientific and objective analysis based on experimentally proven data will be essential to address precautionary actions to prevent or minimize the damages to the nature. Finally, I thank the organizers of the UNESCO and Tata Institute of Fundamental Research for inviting me as a guest speaker of this event held to celebrate the World Environment Day 2023. I plead everyone who lives on the earth today, please contribute in whatever the way you can, small scale or larger scale, to make the Mother Earth a safer place for future generations. Heal the world forever. Ayubowan.
Thank you, Dr. Kokila, for a wonderful explanation of laws and in the context of nature, environment protection, sustainability, and the role of sustainability by giving very wonderful examples from various places, particularly Asian countries, where laws are there, but we, as a common man, we are not aware of many laws. It's a really very nice and informative talk, and the role of basic sciences in a legal aspect, which you elaborated very nicely. Thank you once again. Now I hand over the sessions to coordinator, Professor Arnab.
Yeah, can you stop screen share?
Yes, sir.
Okay, we are doing really, really well on time. Thank you, all the speakers. You know, this has been excellently organized. Can you stop screen share, please? Sonali or whoever is doing it. Yes, great. Okay, so now let me invite Bianca. It is time to figure out what happened to the periodic table challenge. And I must be honest, I myself have been playing with the periodic tables. I mean, I just love chemistry. I love the periodic table. Rhenium is my favorite element. Don't ask me why. Anyway, so Bianca, how has the response been to the periodic table challenge today? And do you have some interesting news for us?
Everyone. So for those who don't know me, my name is Bianca Davis, and I'm the Vice Chair of the International Young Chemists Network. On behalf of IPAC, we'd like to thank all of those who've participated thus far in this year's challenge. So since its inception in 2019, we've had players from over 160 countries. Wow. And this morning, we encourage you to participate, as well as encourage your colleagues to take part. So far, we've seen players from over 4 countries, with players answering more than 70 questions correctly. But there's still a lot of time ahead of us. So we'll be providing multiple updates on the challenge as the 24-hour progresses. And just a reminder, we'll be 3 randomly selected winners tomorrow morning at 4:45 UTC. Thank you so much.
Thank you, Bianca. Thank you so much. Okay, so then let me try and summarize today's session before we wrap up. It's been an amazing 4 hours. What should I say? I mean, it's very rare to be able to sit on a Zoom call for so long, but this has been just so much fun. I mean, we started off this morning, well, this afternoon for us, morning for some of you in Europe, perhaps, or wherever you're watching from, whichever time of the day it was. We did a historical journey through the life sciences and how basic sciences itself has been a driver for a lot of research in biology that has led to advancements in medicine. That was from Professor Shashidhara at the National Center for Biological Sciences in India. And then we sort of changed gears and moved on to the panel from the International Union of Biophysics, and Christina gave us an introduction to biophysics and how it was connected to global sustainability and health. And we had 4 excellent presentations. Matthew in Oxford told us about how structural biology helped in designing vaccines for malaria. And from Oxford, we went across the world to Thailand, where Pim Chai told us on a very different topic of using enzymes to get agriculture more sustainable. And then we moved back to something we've been always worried about from COVID, which is respiratory medicine. And Jesus from the university in Madrid related the research in his group in biophysics to understanding respiratory medicine better. And finally, we had Miguel from Portugal, who decided to look at this very, question we have in our minds about, can we be prepared for a pandemic by the next pandemic, whenever it comes? So we had these 4 lovely talks, and then we went back to something that connects to today, Environment Day. And we started off from the beautiful island of Mauritius, where Dr. Shakil connected STEM, STEAM, and all the basic sciences to the idea of environmental sustainability. I am sure it's something which is very, very relevant for island countries. And we took a break then away from English, moving to a small session that was done in the last week at local colleges and schools in Maharashtra, in a small place near Satara, by Sudhir Kumhar and Narendra, who dealt with this very contemporary topic of plastic pollution and how students understand it and what students feel about what they can do about it. And finally, we just heard Dr. Kokila from Colombo, who essentially asked a very interesting question about, does nature have rights? And what is the legal perspective about looking at these environmental issues? So it's been a great mix, fantastic speakers, very, very a lot of things to learn today. So first, I'd of course like to thank all the speakers. In particular, I should thank the Secretary General of the International Year of Basic Sciences for Sustainable Development, that is Luc Allemont. You probably saw him very briefly on the video explaining how the periodic table was supposed to be, the game was supposed to be played. Luc has been absolutely critical in driving this show and getting us all together, making sure that everything fell into place, arranging all the Zoom links, YouTube connections, and everything else absolutely perfectly. I'd also like to thank Narendra, Sonali, and the team at HBCSE who've taken all the effort to make sure things are happening well, and Ulhas at TFR, who's been working on some of the technical issues behind the scenes. So thank you all.
Can we take a snap?
Also enjoyed a good time, and let's get some words from Narendra before we end this session for everyone. Go ahead.
Just can we take a snap if everyone we can use the camera?
Okay, let me remove my spotlight. Let's see if people are willing to turn their cameras on, and then we can take a shot, of course.
Ulhas,
go ahead if you can.
Ulhas, Vandana ma'am.
Yep, Ulhas is there. Ulhas is bended so that you don't see the ceiling.
Oh yes. Ulhas, can you take a snap?
I can do that.
Okay.
I can do that. New.
Vandana ma'am, screen चालू करा. Camera चालू करा.
There we go. Okay, we have one more person coming in, which is great. We'll take another one. Fantastic. Thank you so much, everyone. Any last words, Narendra?
No, sir. Thank you. It's a wonderful sessions, a wonderful slot. We tried our best. Let us celebrate World Environmental Day.
And let's hope that basic sciences that we all do goes ahead and gets supported. Yeah. We've been very lucky to be supported in India, but I hope we do support basic sciences because that is the route to find the solutions we need to save the world in the future. On that note, thank you, everybody. Thanks a lot. Enjoy a nice day, evening, night, wherever you are, and stay tuned. Bye-bye for now.